Perfect. I think we are already recording. Once again, good morning, everyone. We will now begin this workshop on best laboratory practices organized by Minor Use Foundation with the support of the Inter-American Cooperation Institute for Agriculture, IICA. I'm very much interested in providing some logistic information that can help us have the best event possible before handing the floor to Anagor for opening the event. It's important that you realize that this workshop is for participants in Latin America and Africa. This is why we are doing the event in this manner. We thank you for your participation, especially those who are connected at a very unconventional time. I'm going to begin with the instructions of the buttons of interpretation and language selection. So I thank you for sharing the PowerPoint that shows us these indications. The event is in 3 languages, English, Spanish, and French. There are 2 buttons in the platform, the Zoom platform, that I hope you can manage properly. And this is for you to hear in your language of preference. As we saw in the video, in the bottom, you will be able to see a button called interpretation. And clicking on that button, you'll be able to select the language you want to hear. So I hope that right now, everyone has selected the language of preference. For those of you who are presenting, please be consistent with the channel where you are and the language in which you are speaking. If you present in Spanish, you should be connected in the Spanish channel. This is important to avoid any echoes, distortion that are bothering for the participants. And all the presenters, please speak slowly. I remind you once again that we have simultaneous interpretation, and if we speak too quickly, the interpreters will have more difficulties in providing a more truthful message. The presentation of the PowerPoints will be done by Adriana in English, Maria in French, and me in Spanish. Therefore, I request all the presenters to select the following word every time. Oh, say siguiente when you want to go to the next slide. That's when we'll know how we can move forward in the presentation. The other button that's on the top of your screens, view options. Once you click on that button, you'll see the 3 options of presentation in PowerPoint. You'll be able to see the presentation in Spanish, English, or French. An important point you must consider is that each time we change a new presenter, for example, the language selected to see the presentation of PowerPoint might change. So if this happens, go back to the button and select the language in which you want to see the presentation. The event will last 4 hours. We'll have 7 presentations today and 6 presentations tomorrow. There will not be Q&A sessions or comments after each presentation. We will do this at the end of the event, and we will have 20 minutes to answer as many questions as possible. Please, if you have any questions, any comments, go and write them down at that time in the chat. This will allow or will facilitate answering these questions. I remind you that this event will be recorded and you will have the possibility of having this content available on the website of the foundation. For when you want to look back into this training session or recommend this to any other colleague. For the next 2 days of workshop, we have 4 presenters from the foundation and 6 presenters from countries that have implemented these essays. And this is an opportunity of having real experiences on the good laboratory practices, GLPs. We could consider that these delegates represent the first generation of specialists that are official specialists with experience in these matters. You might have seen that we have no record of the participants. Therefore, we no registration, sorry, of the participants. So throughout the event in the chat, you can write down your name and the country you belong to. This is valuable information that will help us map the participation of everyone today. I don't want to use more time with this information. Therefore, I am pleased to present Anna Gore, who is the Director of Operations of Minor Use Foundation, and she will give us our welcoming words for this event. Please, Anna, you have the floor.
Muchas gracias, Eric.
Thank you very much, Eric, and welcome everyone.
I will speak in English today since I'm in the English channel, just to make sure that everybody can understand. I'm really pleased to open this session. I think this is a really exciting meeting. We don't usually do meetings across regions, so I'm hoping that there can be some good learnings from one region to another, and some good information shared. And we have some excellent experts that are going to share some really valuable information on good laboratory practices, which is a really important issue. In order to be able to do the work that we do in field research and establishing codex MRLs, we have to have good quality. Sorry, my cat is coming in the picture. But we have to have good quality laboratory work, or else we don't get the results we need to be able to do our work well. So this is an important topic. I'm so glad that you could all be here today. I know that you're going to have a very successful meeting over the next 2 days. And after this, as Eric mentioned, you can find all the presentation information available on the Minor Use Foundation website. We will share that link with you. So I wish you the best for a successful meeting, and thank you for being here today.
Muchas gracias, Ana. Y bueno,
thank you, Anna. And I think we can begin with today's session. As I mentioned, it's going to be very interesting, 4 hours with an exchange of experiences. And to begin, I have the pleasure of introducing Estela Semillo. She's a consultant from the foundation. She will present the construction of capabilities for study of waste and residue in Africa. Stella, you have the floor. Thank you very much.
Okay, thank you very much, moderator of the session. My name is Stella, as has been said. I'm from CropLife Africa Middle East. I also help the Minor Use Foundation to coordinate the work going on in Africa. And I'm pleased to be here today to provide some perspectives on the capacity building that is going on in Africa. Next slide, please. Thank you. We cannot speak about the capacity building work that is going on without putting it in context to just appreciate what other challenges that the experts from Africa face in terms of agriculture. We are happy to join the workshop collaboratively with other regions, and I hope that sharing our challenges and sharing our experience will enrich the work going forward. So in terms of the countries. There are many countries, of course, at different levels of development, but all of them face in climate change, and this causes a lot of brings about the pest and disease pressures. You have also heard about the drought situation that affects our region, and sometimes most of agriculture is dependent on rain, and some of the challenges therefore that farmers face and the experts here face when they are supporting the farmers, technological access is a major one. And this cuts across the regulatory frameworks in terms of regulating the technologies that are available. It cuts across also the issues of access or availability, affordability issues. And this affect the production of agricultural commodities. Another challenge is the market access. I think I will not belabor this point because most of us in this platform would understand the issues that are faced by both regions in terms of accessing some of the critical markets. And then increasingly we have consumer safety concerns. And when you talk about residue, of course, the export market, because of the standards that have been put in, this is well catered for, maybe beyond 90%. But when you look at the local market, for which almost 90% of the products that are produced by the farmers remain in the local market, there are concerns around safety. And this is a discussion that is increasing in intensity. And then we have another challenge of changes in policies from other regions, especially the focus markets like the EU. And this is a big challenge for a farming community, especially those that participate in export. And then overall, the low public expenditure, which also affects the issues around capacities. How many experts do we have in this area? Are they supported from the public budget? The infrastructure that is needed for residue studies is also a challenge. So this, in summary, are some of the challenges that provide some context in understanding where the region is coming from, especially when they participate in this collaborative research. Next slide, please. Next slide, yes, thank you. So what is the rationale on the action that is going on around minor crops? I think this is understood by all the experts here that there is need to overcome the trade barriers, some of the challenges that I have mentioned, and increase trade. By so doing, enhance the livelihoods, which is the income that comes from the exports. And a very interesting statistic is that up to the contribution of minor crops, the income or the revenue that comes from minor crops or minor crops, specifically fruits, can account to up to 75% of the entire income of small rural households. This is really interesting for us. And why? Because most of our farmers producing minor crops are smallholder farmers coming from rural households, and therefore it makes this area of research work very critical for our region. Next slide, please. So I thought to share some perspectives from past interactions with African country representatives on discussions around MRLs and minor crops. And this speak to one, the rationale, and it also speaks to the challenges that are faced around supporting the minor crops research. I will not read everything. I'm sure the slides will be available, but I'll just highlight some of the ones that have been bolded. In terms of MRLs in export destination markets, some of the limitations that the African country representatives cited were the lowering of MRLs in export destination markets as a limit, the absence or the missing of MRLs for specialty crops, or if you like, minor crops. And then the issues around cancellations of products or active ingredients that are used on these crops in some of the destination markets brings about restriction for exports from the region. And then the different requirements in the different markets have also included sanitary and phytosanitary problems. And some of the proposed actions from the representatives who are engaging at international meetings, or including the SPS, WTO meetings, strengthening the codex implementation and setting of codex MRLs. And the question that the participants kept asking themselves is, how do they ensure that there's active participation and engagement in the codex MRL setting process? And this brings us to the central objective of the capacity building work that is going on in Africa on residue studies. To ensure their active participation, it means that they need to participate in generating data. And so this was one of the areas that was highlighted, that there needs to be sustainable training in data generation to submit data from Africa region to support the setting of MRL. You will all recall that African countries differ to the Codex MRLs. Next slide, please. So what has been the involvement of Africa, its experts in the residue work over time? I will stretch back to 2012, where there was the Africa Union Africa Pesticide MRL initiative. In between, there have been other initiatives and meetings, and the one I will highlight is the residue data that was generated by some of the countries represented here: Senegal, Kenya, Ghana, on the mango trials on sulfoxaflor. And the good news is that because of the involvement, we have an MRL recommendation for adoption in this coming CCPR meeting. So this really is an encouragement of the fact that the involvement of different countries and the different experts is very beneficial for this process. And of course, it helps to have the different organization, IICA here, and facilitation by the Minor Use Foundation, to focus on the minor crops that most of the tropical countries are producing. So I wanted to highlight this because it forms a point of encouragement that the work that is going on in the countries is critical, not only for the farmers now, but we are sure that more farmers will be engaged, because there are solutions that these experts here are supporting. So in terms of the process of the research, collaborative research, and this, forgive me for those who are already in this process, But for those participating for the first time, it starts with the creation of the study teams in the countries. Then it is followed by highlighting the crops, the priority crops. Of course, this is a guided process, and the identified pesticides to be used on that crop. Then it is followed by field training, laboratory training in GLP, and I'm happy that we are having the first training this year. Then the conducting of actual research, followed by data review, and then data preparation and submitting to Codex. It's a process that involves many of the experts here from the Minor Use Foundation and other research experts that the Minor Use Foundation brings on board, and really to help then streamline and make efficacious the process of generating residue data. So this I wanted to share as a highlight for encouraging our region, but also other researchers that are joining the teams. Next slide, please. So what are the current projects happening now? In Uganda, we have a coffee trial. I think it's finalized now, and we have also the passion fruit trial. This is for 2021-2022 period. And then in Kenya, we are set for the fruit trial on passion fruit trial on sulfoxaflor as well. And the other countries are also coming in. You realize that we have 2 countries now participating, but on the call and in the research teams, we have about 7 countries now that are planning to participate in the different work that will be slated. So this is an encouragement that soon we will have on the map more countries and more trials this year into next year. Next, please. So what are we doing in Africa? We are working on expanding the collaboration, meaning more involvement of countries. And we are looking to have the one shaded in amber. We are looking to have Egypt, Ethiopia, Morocco, Cote d'Ivoire, and South Africa join in at different points. For some of them, we have initiated discussions. For some of them, we are yet to initiate the discussions. Currently, we have engagements with the country experts from Tanzania, from Malawi, from Rwanda, from Uganda, from Kenya, from Ghana, and Senegal. And this is aimed at addressing the challenges that farmers are facing around minor crops, the issue of missing MRLs, and building capacity for more people to participate in residue trials. We know that these are expensive, and these collaborative efforts, learning from each other, and in the last trial, some countries were sharing labs and transporting products for analysis, samples for analysis in other labs. So these collaborative efforts are helping for the countries to conduct residue studies. And I'm hoping that with the coming in of other countries, there will be joint experience, there will be experience sharing and just helping each other where infrastructure may not be sufficient for another country, or maybe experts meeting together and helping to analyze the data, or helping to make efficient the trial that will be going on. So I'm very grateful for the discussion, and I hope that this continues region to region, country to country, and this will help in improving the efforts that are going on. The next slide, please. So other activities that are in the horizon in Africa is the Africa Regional Priority Setting Workshop that is slated for 11th and 12th of October this year. There will be preceding activities which we'll be sharing with the African participants, experts, to be involved in and engaged in, and the outputs from this workshop in October will be used as part of the discussions in the Global Minor Use Priority Setting workshop that will be held next year. We also have the implementation of the East African Community Guidelines on residue studies going on, and this also is a resource to be used by countries within the East African region, but also some ideas can be borrowed by other countries that are participating. And we also envisage to have one more targeted training for the country teams, either country specific or joint. So these are the activities slated for this year. And as well, we have the update meetings that happen every month, and this will be communicated. With that, I would like to thank you very much. We can move to the last slide. To thank you all very much. If there are any questions, you may need more details, please put in the chat and we'll be able to handle this during the session. Thank you. Thank you.
Muchas gracias, Estela, por esta muy interesante
Thank you,
presentación.
Stella, for this interesting presentation with respect to the experience and the path that you have had in the African continent building these capacities for residue studies. Next, we will go on with our next presentation today, which is Joe De Francesco, doctor and consultant of Minor Use Foundation, which will be talking about good laboratory practices. Please, doctor.
Thank you, Eric, and good morning and good afternoon to everyone. It's a pleasure to be here. This morning, I'm going to be discussing good laboratory practices in general, what they are and why they're important. As you can see from this slide, that I and Ken Samoil, who is also a consultant with the Minor Use Foundation, put these slides together, and we are located in the United States. Next slide, please. So as I said, this is a very basic presentation on exactly what GLPs are, good laboratory practices. We refer to them as GLPs, and why they are so important in residue studies. Next slide, please. So before I get into the details of good laboratory practices, I think we all need to remind ourselves that it's good science that produces good data. And with pesticides, with any chemicals, the evaluation must be based on test data of the highest quality and reproducibility. And to that end, good laboratory practices promote the development of quality test data. So if we are going to have good data for residue studies, we need to conduct them using GLP guidelines. Next slide, please. So what exactly are GLPs? Well, GLPs are a set of guidelines that cover the organizational process and the conditions under which studies are planned, performed, monitored, recorded, and reported. So basically, all aspects of a residue study are covered under the GLPs from the very beginning, how the organization of your organization and facility is set up, and the conduct of the study in the field and in the laboratory. Next slide. So a reminder that GLPs are applicable to both the field and the laboratory, even though the name is good laboratory practices. these guidelines do apply to the field portion of residue studies. And so the GLPs promote the quality and validity of the test data and the data that's used, all that data in the residue study that's used in determining the safety of the chemical products, which are the pesticides, and the commodities, which are our food commodities. So basically, GLPs help ensure uniformity, consistency, reliability, reproducibility, quality, and integrity. To be able to reproduce a GLP trial, that there is enough data documented is very important. You want to be able to have enough information in your field notebook and your laboratory notebook that someone coming in from the outside and not fully understanding the project can be able to tell exactly what went on and who did what and where the data came from. So all these aspects of a study, uniformity, consistency, reliability, reducibility, quality, etc., are very important, and the GLPs help ensure that the study will be producing quality data. Next slide, please. So what are they exactly, and where do they come from? The development of the GLPs were developed by the Organization of Economic and Cooperation and Development, which is an organization that was founded back in 1961. 38 countries got together and decided that they wanted to create a set of guidelines to help ensure economic progress in world trade. Excuse me. The OECD guidelines presents a set of standards that are used and accepted internationally. And as you are learning today, all minor use foundation studies and most international residue studies adhere to the OECD GLP guidelines. So learning about them and abiding by them are very important as you participate in residue studies. Next slide, please. So the guidelines are quite extensive. I think there are about 47 pages to the guidelines when you pick up a copy. But I wanted to give you a few examples of the types of things that the guidelines cover, or the topics that the guidelines cover and ask you to do as a researcher. So to begin with, the study plan, which is what we call the protocol, has to be reviewed and signed by the sponsor, the study director, and the quality assurance unit. Now, you'll be learning more about these roles, the sponsor, study director, quality assurance, later on today and tomorrow. But those are people who are involved with the study. And a study is not officially initiated. It's not official until the protocol is signed. So getting those signatures are very important so the trial can officially begin. Another example is that equipment, instruments, and devices used in a study, and that's both in the field and the laboratory, should be periodically inspected, cleaned, maintained, and calibrated. And not only that, but a record of those activities should be maintained. Documentation in a residue study, according to GLP, is imperative. We have a saying that if you did not write it down, it did not happen. So documentation is very important. Anything you do in a residue study needs to be recorded in the field data notebook or in your laboratory notebook. Again, this is so reviewers of the data can see exactly who did what and where it was done. So remember that you can never have too much documentation. It's always better to write and explain too much than not enough and leave people guessing. Another GLP topic deals with the test substance, which is the pesticide in the field or the analytical standards in the laboratory. They've got to be labeled properly, stored properly, and temperature monitored. When you're using these products in a study, if they're not labeled properly and kept under the correct temperature, and it's documented that you've recorded that temperature, there's no way of knowing that that product has not been compromised by too hot temperatures or maybe too cold temperatures. So taking care of that test substance, making sure that it's stored properly, and you have a temperature record of the temperature where it is stored at all times is very important to record. Another example has to do with recording of the data. It is not permissible to record data when you feel like it. The data should be, that in a study should be directly, promptly, accurately, and legibly entered by the individual who's entering that data. So it must be done in dark ink. A pencil is not allowed. Crossouts, if you make a mistake, which of course is normal, is bound to happen, but you can't erase or black it out. You must put a single line through the error and then initial and date that error, put the correct information there, and make a note of why or how that error occurred. That way, again, someone reviewing the data can see that maybe you wrote the wrong number down or you wrote the wrong information in the wrong place. And they do not have to guess why there was a mistake and how you corrected it. Next slide, please. I will look at my English example. And why are they important? Again, I hear some static, but maybe that's just on my end. So those are what GLPs are, but why are they so important? Well, one of the main reasons is that national regulatory agencies, authorities in your country and around the world, for every country, those agencies that assess proposed pesticide registrations, they require that the supporting data be conducted in accordance with these GLPs. And as we know, quality data forms the basis for mutual acceptance of data amongst country. So if a country from one region is exporting to a country in another region, knowing that the study and the MRL was based on GLPs, they have insurance that the data is good quality data and that when your product is shipped to their country, that the pesticide levels on that commodity have met international standards and they're at an acceptable level. So GLPs are important not only to ensure quality data, but they do help move product around the country and around the world. Next product, please. Next slide. Another reason why they're important is the principles of the GLPs, these guidelines that I'm talking about, help to avoid the creation of technical barriers to trade, just as I was explaining, knowing that the data was collected under very strict guidelines, gives countries confidence that the commodity will be safe for their citizens to consume. And GLPs also improve the protection of human health and the environment also. And lastly, it's not the only last reason, but another reason why they're so important is that the data that you
En este momento no estoy escuchando la presentación de Joe y me parece que quedó congelado en la imagen. Probablemente ha tenido algún tipo de problema de conexión.
There's possibly a connection error for our presenter. We will wait for a couple of minutes and we will continue as soon as he's back.
Yeah, I'm sorry. I lost my internet connection. I think that was me. The last thing I was explaining was that, and they're in English now. Thank you, Adriana or Veronica. To establish an international Codex MRL, which is what they absolutely require the adherence to GLP guidelines. So they're very important for the projects that all of you are working on or will be working on. Next slide, please. I'm getting to the end here. I just want to say that you'll learn more about GLPs over the next couple of days by various speakers who have experience. You do know that when a study is conducted, you will have a local quality assurance person in your particular country who will conduct audits of you in the field and you in the laboratory to verify that the GLPs are being adhered to. And don't forget also that your local QA person is a person to ask questions of, but those of us here at the Minor Use Foundation, myself, Ken Samoil, and many of us with the Minor Use Foundation are here to help you and assist and address any GLP questions you may have. Next slide. I want to thank you for your attention. I apologize for the loss of the internet connection. And as Stella mentioned earlier, we will be taking questions at the end of the session. So if you have any questions, please don't hesitate to ask. Thank you.
Muchas gracias, Joe, por esta
Thank
interesante.
you, Joe, for this interesting presentation about good laboratory practices. Maria, I would like to think maybe if you could stop sharing the screen in French.
Listo, perfecto. Gracias, María.
Great. Thank you, Maria. Next, we will have a series of presentations when they will be coming on with officials that recently were making their first steps in this processes and began with the educational process. And it's very interesting to see now they have become educators themselves and officials that can transfer their knowledge and experience in their own countries, in the region, especially, and even with other continents. This makes us all very proud as a foundation because we see the fruit over time and we begin to visualize and to really see up close the sustainability and the impact of the foundation's work in our own countries. Before I go on with our next presentation, I think we have the integration of other delegates. And I would like to remind you that you have two buttons in the Zoom platform, which are very important for the good use of the platform and a good follow-up of the workshop. In the bottom of your screen, you can select the language in which you want to listen to the presentation, English, Spanish, or French. According interpretation channel, you can pick that language to be able to hear the presentation in that language. And in the top of the screen, there is an option which are view options. When you click on that, you can visualize three options among which you can pick the language in which you want to see the presentations as they are shared on the screen. In some cases, the system may reconfigure, or every time that we change a presenter, we go back to sharing screen, and maybe you can see on the screen a visualization of a presentation on a language that's not the one you want to see. So please remember, if you change the language of the presentation, simply access the top of the screen with view options and choose the language that you wish. For the new people coming in the last couple minutes, we give you a very warm welcome. We will go on with our next presentation for today, which comes from Saul Flores, consultant of Agrocalidad in Ecuador, and Ken Samuel, consultant of the foundation coming from the United States, and they will be talking about organization and staff for trial installations. The floor is yours.
Saúl, si estás hablando, creo que estabas en mute.
So you're on mute. We cannot hear you.
¿Confirma si pueden escucharme allí?
Ahora
Can you hear
sí.
me now? Yes, we can hear you now.
Saqué mejor el micrófono. Buenos días, buenas tardes a todos. Mi
Good
nombre
day,
es
good afternoon, everyone.
pertenezco a la agencia
I am part of the regulatory agency of agroquality, which is a government institution in Ecuador, which is a focal point for cooperation with the initiatives of minor use. Now we will go on to the topic at hand, trying to explain briefly. Please, can you confirm if everyone can hear me? We can hear you, yes, Saul, and the presentations are on screen. We will try to translate the content of good laboratory practices in what is applied to our trials. The information that we'll be sharing makes reference to good laboratory practices in the Organization for Economic Development, so you can have access to these documents and be able to understand in detail the summary that we will be showing today. Next. Very well. Only to remind you, we have 10 principles, as I mentioned, and we will be speaking about the first principle, which is where we begin the planning stage, and that's the organization of the staff. It's one of the primary stages, not slightly taken in the first place, but do not feel overwhelmed if there's a lot of information, and you can corroborate those that have gone work with the foundation before. These are matters that start being clearer at the detail level, but it's something that we're required to be able to fulfill good practices. And this presentation will be divided, and we will be presenting with Ken with certain parts of the presentation. Next. Very well. Just a minute, please.
Ok. La organización y el personal.
The organization and the staff,
Hay un personal mínimo.
there is a minimum staff that is required to be able to fulfill or say that we're fulfilling good laboratory practices. In this case, the staff indicated by the guideline at least should be the following. We need to have a test facility management, a study director, a principal investigator, study staff, quality assurance personnel, and management of archives. This is all present in the document. The 6 categories that we have on screen are primordial to be able to give that international validity to the study among different institutions that we have maintained, and we are performing good laboratory practices. Now, I will try to explain a small extract of each one of the roles on screen. Of course, we will not be able to go too much into detail, but it's also a good idea in the recovery of the study, but we'll basically translate this into the application setting in the implementation. Next. Very well. We'll go directly with responsibilities. And this is the maximum authority trying to have a primary it'll try to fulfill every single principle of good laboratory practices. It is in charge that the principles in the guideline are followed throughout the study, and if there are modifications, which will be seen afterwards, these never are far from good laboratory practices. It's very important to verify the existence of a document or a registry where we assign the people that will be working in the trial installations, their responsibilities, and everything according to the guidelines of the study. Also, they must verify that there is enough of a number of qualified staff and personnel, equipment, devices, and everything has to do with the adequate materials for the trial itself to take place. Also, they must have a control or a file that registers the different qualifications and the experience, divisions of tasks of all the different people participating at a professional level. And also must look for all the people involved to be able to understand what is the responsibility. Also, trying to allow for enough standardized procedures to be able to maintain good practices. The more normalized procedures and operational procedures we have, then the easier it is to reproduce these trials independently of where they are. In the same way, you will see this when we go into practice and when you start collaborating with the foundation, that there is a program, there is a team to ensure quality, and basically gives more formality to the fact that we have respected good laboratory practices. Next. In the same way, one of the biggest attributions when picking these installations is to assign who will be the director's study of these trials. And once this person is assigned and we're able to assign the ideal person, then to work with this person in the planning of the trial and to have a roadmap that's being understood thoroughly by the field investigator. In the same way, we will need to verify that the study director has given the resources for the elaboration. And when we go on to the next slide, the question primarily is how do we manage to be able to fulfill these minimum requirements? Next. Very well. So what is it that we need? Minimally speaking, indispensably speaking, we need to elaborate an organizational chart, have a job profile, acceptance of responsibility, a job program, and a quality assurance team not involved in the study. We'll need to revise this quickly. Next, please. Very well. Here on screen, we see an example of an organization chart. And you can see on the top, you have the test facility management, and then you have a study director, then you have the people in the quality assurance unit. And the study director also has a communication from the people in the foundation. And under the director's study, you have two primary stakeholders, one the field investigator and the other one is the laboratory researcher. And both of them also have supporting staff for each one of their activities. One thing that is very important to highlight is that if you notice, the test facility management does not respond directly to the study director, even though there is a constant communication. I want to give you an example of what we have done in the case of Ecuador. Most likely, not always the case, the countries cannot have in one sole country the replicants of a trial. Sometimes these trials must be developed in collaboration with other countries. For example, Ecuador added to this initiative that's taking place in the case of Colombia with respect to trials in pineapples and priority options that we have investigation.
Given that all of the staff was in Colombia and that we did not have the sector or area where the crop pineapple to be analyzed is not in the same geographic area of the capital where the main offices of the agency are. What happens there is that we decided that, as you see here in this org chart, there can be an additional role, which is the one of the local coordinator. The local coordinator that operates as a connection, as a bridge between the study director and the field investigator. It allows the field investigator to have all of the necessary tools to meet the protocols. So, for example, in this case, the agency itself has its main offices in the capital, but the agency has different technical branches in all of the provinces. When the country has determined what crop, what combination will cooperate, that's when the local coordinator, in this case, I've tried to meet that role, has to be in charge of being that bridge between the field investigators and the study directors. And likewise, that facilitate that we have every necessary stakeholder for all of these trials. We've determined, for example, that we'll participate in mango, for example. So the local coordinator has to seek all of the stakeholders, be a bridge of communication between the people in the territory and the possible different farmers that can participate, and allow to have a discrimination where uh, of them being technical partners or participate in this. They also are in charge of receiving all of the necessary information from the study director to have everything ready. This in terms of the needs that have to be met by the field investigator. Let's say Ecuador started last year with these trials in Santo Domingo, a province that is about 3 hours away from the capital. We purchased equipment. They have to be gauged before being used at the field. And this gauging has to be a contribution that allows that cannot be a responsibility of the field investigator, because companies that do this calibration, this gauging, weren't in the capital. They were in other different points in Ecuador. So all of these logistics organizations, all of these details, what we seek is that the local coordinator facilitates this so that the field investigator, just as time passes by, and you'll see this tomorrow, they have several different responsibilities. They should have the facility of doing their own work. It's mostly mandatory when the country cooperates with a study director or a facility direction that is not within the same territory. Next, please. Now, the next point is, as I said before, there has to be an acceptance of responsibility. What you see here on the screen is an example of how we manage the designation. There has to be a formal designation with the names and names of the roles and of the officials, specifying what trial they're going to be working on. This is just an example. On the right hand of the screen, you can obviously see that each institution has their own document management system where they manage all of the letters, memos. And the message here is that there always has to be a level of formality in these allocations, these designations, and it has to be recorded. There has to be a way to certify that the person that's working knows the different instances in the trial and that they were designated on time and properly. Next, please. Sometimes, sorry, we also have another requirement, and that's to have a work plan. The work plan has to be approved by the study director. And further on, you'll see that there's many different formats that are saved and executed within the different GLPs and these trials. Basically, what we have to do is a schedule of how the activities will happen. The different formats can be managed according to the different institutions. And this is an example. In this case, here we have a schedule for these trials. The first one is already done last year. And basically, what this says is these are the activities that I'll have, and the activities I'll have for that trial, the dates, the days, the weeks, and also very important, and this is just to confirm, on the right, we have the day intervals, because depending on each protocol, we have to respect certain distance between these days. So what you see here is basically when we start and the dates that followed these applications to meet the work schedule itself. So each trial has this, they should have it. And here we also have the local coordinator working hand in hand with us to be able to better convey the information we find at the field. If the study director is in another country, for example, they can't know firsthand what the conditions are in terms of climate, the road conditions when we can't go there on plane. So all of these details are what the local coordinator helps with so that there is some sort of fluency. And the schedule you see here on the screen should also be met as strictly as possible. That's what they also make sure happens. The next portion is a detail of the next position, which is very important within the trials that will happen. with the foundation. The main message is that if we haven't been able to cover all of the information, these are things that you'll learn on the way. Don't worry about that. In progress, so to speak, you will continue to learn about this. The important point here is that everything has to be recorded. Everything has to be set out in a document. And when there's communication with the study directors, we have many formats that have been established with the minimum information required. So if we keep in mind that we have to have a strict and continuous record, then it's going to be easier to see what we have and we have filed and archived. The next part is what corresponds to Ken. He will explain the responsibilities of the study director. Thank you. So Ken, you have the floor.
Thank you. Hello, everyone. I'm Ken Samoil, and I have been a study director for many years, mostly in the United States, but more recently with the Minor Use Foundation, and I shall deliver my remarks in English. May I see the next slide? The OECD GLPs define the role of the study director as the single point of study control. The study director has responsibility for the overall conduct of the study and for its final report. The study director must approve the study plan, which the foundation usually refers to as the protocol, and I will use that term, but they are exactly equivalent. The study director must approve the protocol and amendments to the protocol by dated signature. And the study director must ensure that all of the study personnel and the quality assurance personnel have a copy of the protocol and amendments. Another responsibility is to ensure that the protocol and the final report define the role of the field research investigators and the laboratory investigator and identify the testing facilities. The study director must ensure that the procedures specified in the protocol have been followed, assess and document the impact of any deviations on the quality and integrity of the study, and follow up with corrective action if needed. And I'm going to pause here a bit. I think this is the most important part of the interaction between the study director and the field personnel and the laboratory personnel. If you have been assigned a field trial and you are looking at a draft protocol and what it instructs you to do, and you know that for some reason you will not be able to follow through with those instructions as they are written because of the timing, because of the equipment needed, because of the resources, the personnel you have available to help you. Or if you are in the middle of a trial that is underway and something happens that will prevent you from being able to carry out the instructions because of a weather event, because of some emergency involving the people you have, or because of an equipment breakdown. You may have several good ideas as far as how to finish up that trial and harvest a crop from the field that you would be able to sell at a farm market if the chemicals you were applying were registered. There may be a few different ways that you could successfully complete the work, but it is important that you contact the study director to express your concerns about why you can't follow the instructions as written, because not all of the ways to get a good harvest would produce the data that is needed to support the proposed pesticide use, the maximum residue limit. And so it may be that you will end up with a good harvest of fruit, but the trial must be terminated because the data cannot be used to support the use because of the change in timing or the change in equipment or some other reason. And the study director will know which options are best to be able to get data that might not be exactly what was intended originally, but will still be close enough to be useful for this purpose. So especially if you are looking at a draft protocol and something doesn't seem right, please contact the study director to explain your concerns. And if the trial is underway, if there is time to contact the study director in advance, again, please do so. And because we're at a great distance, that will usually be by email. Sometimes you have to make a decision on the spot and do what you think is best because there is just not time to wait until you collect the fruits from the field, or there is just no possibility of delaying action. In that case, you must prepare a protocol deviation form and submit that to the study director, who will then assess whether your actions have will still be supportive, will still be useful, that the trial can go on and the samples will be analyzed. And all this is true for the laboratory personnel as well. If anything cannot be followed according to the protocol, the study director needs to be involved to ensure that whatever the alternative action is followed, that it will still result in data that can be used to support an MRL proposal. The next responsibility of the study director is to acknowledge any deviations from standard operating procedures during the conduct of the study. So if you have any deviations to your own SOPs, please pass them on to the study director because that is part of the responsibilities. Usually those are not critical to completion of the trial. The study director must ensure that all raw data are fully documented, and that is something that we are largely reliant on the local coordinators for. But if it appears that the data are incomplete, then the results of the trial would not be useful in an MRL proposal. The study director, according to the OECD GLPs, must ensure that computerized systems used in the study have been validated. Now, in the field trials, I don't think we use any computerized systems. The analytical laboratories have some equipment that need to be validated, but because the study director in a magnitude of residue study is usually very remote from the laboratory, we are largely reliant on the quality assurance on site to determine that the systems are validated. I should note here that these GLPs are applicable to a wide range of studies. They're not just for magnitude of residue in agricultural crops. So there are many studies in which the study director is on site at the laboratory where all the work is done and can have a more direct role in ensuring, in this case, that systems are validated. The study director must sign and date the final report to indicate the acceptance of responsibility for the validity of the data and to indicate the extent to which the study complies with GLP. And I'll be speaking more about the final report tomorrow. And finally, the study director must ensure that after completion of the study, the protocol, final report, raw data, and supporting material have been archived. In these minor use foundation studies, much of the archiving is done within the countries that the trials are conducted. So I would just, as a study director, need to know that you have archived your own data, but I am responsible for seeing that anything on my end, such as the protocol and final report, have been archived. So that completes the responsibilities of the study director, and I will pass this back on to Saul. Thank you.
Muchas gracias, Ken. Básicamente esa
Thank you very much, Ken. And that basically is what we will meet in terms of the responsibilities of the good laboratory practices in principle. As we mentioned before, you can ask any questions on the chat and we'll try to focus on them and respond at the end of this session today. Thank you very much.
Muchas gracias, Saúl. Muchas gracias, Ken.
Thank you, Saúl, and thank you, Ken, for this very complete presentation of the first principle on GLPs. I can see how on the computer, we're seeing the amount of participants is growing exponentially. We are above 230 participants at this point. So I would like to invite all of those who have just entered, write down on the chat what your name is, your institution, and the country you are in. This is very valuable information for us. We haven't done a registration process, so we kindly ask you to do this for us. Write down your name, the institution you belong to, and the country in the chat. And for those who are just entering, hopefully you have selected the language of preference on the bottom. In language or interpretation, you can select the language in which you want to hear the presentation. And on the top of the Zoom platform, you can find an option which is viewing options. And you can click on it and select the language in which you want to see the presentation. Each time we change a presenter, we can see the option you selected vary. Therefore, if on the screen you see a PowerPoint in a different language, you can simply go up, go to view options, and select the language you wish to see the presentation in. Now, going back to the principles on GLPs, we have next the fourth presentation of today with Veronica Hidalgo from the Phytosanitary Service of the State of Costa Rica with the Program of Quality Assurance Lab and Field. Veronica, please, if you may. Good morning. First of all, I'm not sure if you can hear me very well. We hear you well. I'm going to present in Spanish, so while I open the presentation. As I was introduced, my name is Veronica Hidalgo Madrigal. I'm from Costa Rica, and I work in Costa Rica in the Phytosanitary Service of the State, which is part of the ministry. And we've had a connection with the Minor Use Foundation in some trials we've had in the country. Next, please. I'm going to look into quality assurance, which is called Quality Assurance Unit as well. Initially, to look into this, the objective of the main point of quality assurance is that this unit should inform the test facilities, what we saw with Saúl in Ecuador, on the fact that the studies should have met the principles of best practices, of the good laboratory practices dictated by OECD. So quality assurance can act as a witness during the studies and will be inspecting independently from the study. So the people who work with quality assurance shouldn't participate in the execution of the study. They shouldn't be involved beyond their role and their verification and information in complying with GLPs. This mainly because good laboratory practices should be respected strictly from planning, during the realization of trials, and during the different reports. The quality assurance program also works in the laboratory matters as well as in field matters. And when we process data, this is present in the execution of the trials and studies. And quality assurance, the characteristic is that generally for a region, there can be many regions and locations that have a single quality assurance unit. And each of these regions can have people responsible, and they can act in this way. Next, please. Continuing with a general overview and points that are of significance for the quality assurance program, it's important, as I mentioned, or as they mentioned in the previous presentation, the test facility director appoints this person, and this has to be in written form, it has to be documented, and should be properly archived. And in addition to the quality assurance program, this can be led by one or more people, but these people should have enough experience and test methods. If there's a person, for example, that has enough knowledge on the chemical side for the lab and the field matters, and this can they can have the person for quality assurance. So you can have, as we've done in Costa Rica, we have 2 representatives of the quality assurance unit, a chemical person for the lab and another person in agronomy with experience in the field. As I mentioned before, but I think I'm going to repeat this through my presentation, it's important and a rule that people that belong to the quality assurance program should not participate in the study of these in these studies. So this to avoid an interference. People that participate in the quality assurance program are people that have a direct access and quick at the different hierarchic levels. And so the quality assurance person should have direct communication with the test facility director, the study director, and this to notify and inform the different audits that are done, so that we look into the best practices, to the good laboratory practices. And in case of the quality assurance program, in case it has any anomaly, this should be communicated as quick and efficiently as possible. And the communication and relationship has to be very explicit and continuous in the programming and schedules of execution for each trial, so that the person in charge of quality assurance can be present in the different phases and understand how or when each trial will happen. Next, please. The people who are members of the quality assurance program should read, know, and analyze all of the procedures that have been described and created for the study, with the purpose of each of the parts of these procedures are met thoroughly in the execution of these trials. And these procedures should meet good laboratory practices as well. Likewise, as we saw in the procedures, the study plan and the people who are in the quality assurance plan should have read, understood, and gotten acknowledged with the study plan or the protocol, as it's also called, before beginning the study, so that this person sees that the protocols or study plan agrees with good laboratory practices. Both with the knowledge of protocols and the study plan, they're all important, but it's also very important that the quality assurance unit has controlled copies of the study plan, of the procedures and corrections, or in the case of amendments, then the communication has to be very direct, and there have to be copies that are backed up, and this in a physical form, so that the person who's in charge of quality assurance can constantly look into this and can lead this at any point in time where we're going to have these different institutions. The quality assurance person or assurer has to have an important characteristic, and that's being very organized and very well planned the time guides and the work that will be done, because they have to accommodate to the pace of the study of the trials, so that we avoid any issues afterwards, without this hindering them from inspecting any of the phases that they had planned to inspect. Next, please. The people who work in the quality assurance program have to have certain characteristics and responsibilities. This includes that we have the study plan or protocol be reviewed by the quality assurance unit, so that they meet every principle of good laboratory practices. This does not mean that the quality assurance person will look into the scientific content, but rather what they sign when they sign the protocol in terms of quality assurance. What they're doing is signing off that they confirmed that they are meeting the principles of good laboratory practices. And it's also a responsibility of those who work in the quality assurance program to keep every copy of the study plans, the procedures, the amendments, or any deviation that might have occurred. And this file should be properly saved, or and also with a backup copy. It's also a responsibility of the quality assurance program to show that each of the phases, studies, or trials that are inspected are verified, showing that they are meeting good laboratory practices that have been established. Next, please.
The people that participate in quality assurance must examine the final reports when these reports have finished, and when we are in a final report stage, to evaluate methods that were documented, the procedures, the data registration, how it took place, if they are signed, if they are dated, if there's any type of observation, anything that must be revised. It is important to do this once the report has been done, to quality ensure that there will not be any further data additions. And the idea is that this revision is done finally when we have everything else ready, with the objective that the quality assurance may verify that as a whole, the report reflects the studies and that it fulfills the good practices, and that these reports allow that at some point we need to replicate the study, we have all the necessary data available. It is a responsibility of the quality insurance staff in case that they detect any type of problem or observation in any phase, that the report will be communicated in writing, whether it be an email or physically, in the quickest way possible, showing these results to the study directors and the necessary people responsible of these trials. If we're doing a field inspection, for example, the primary field researcher, if it's on the lab, then we will be talking about the primary investigator in the lab. And in this case, not only should we report the inspections when there was something wrong or an observation or we evidenced an issue, but all the inspections, even though they are positive, must be reported in writing and quickly. In the case of the quality insurance program, it is very important to ask for the people receiving the information, the study director and all the different layers, to confirm that the report was received as quality insurance, to be able to have a clear documentation that the information was received, and even more because it is necessary to write all this down in the final report from the program that we will see further on. It's also responsibility of the quality insurance program, everything that has to do with the files, the copies that were given of the protocol, the procedures, or the original documents of these inspections. Everything must be well documented in a file that is thoroughly protected according to these laboratory practices. Next. Some of the conditions that the person must have to be a part of the quality insurance program, trying to find qualities, certain qualities in people to integrate them into the program. First has to do with their experience and their background. It's important if we are looking at a study and we only have one person that doesn't necessarily have field experience, maybe we should try to ensure that we have quality staff for the lab, maybe with a background in chemistry that knows the procedures, good laboratory practices, but also to try to find a person that goes well with this part of the study being performed on the field and knows about field work. Also knowing how these practices are shared. Not only the background, but also experience is important, that hopefully they have worked in crops that are being evaluated, or at least have a knowledge of what are the equipment is maintained. In the case of the lab, that they know the devices, the procedures, the equipment, and basically that they are familiar with each one of these processes, the trials, systems, and everything that will be evaluated. It is important that they understand the basic concepts upon which the activities are being based, that they have a complete knowledge or background with everything that has to do with good laboratory practices, with a lot of emphasis in quality insurance, so everything flows adequately. It's important that they have continuous education that's documented primarily in good laboratory practices. So this must be evidenced physically and being archived within the curriculum of each one of the members of the program. Next, the quality assurance program to do its job correctly and to find the objective of good practices in the lab during the execution of these studies is constituted by inspections and auditing procedures. So they are a little bit different, but they will take place during the execution of these trials and studies. For example, the inspections try to verify that everything is done according to what was described in the protocol and the procedures. So here we may have 3 examples of inspections, and I'll explain these a little bit more in detail. The inspections relative to the study, and here we're talking about study plans or studies themselves, inspections relative to the facilities. When we talk about the facilities, let's not imagine just a building, but it might be the structures, the equipment, the staff, the files, archives, and also inspections related to procedures, which we also explain this a little bit further, but we inspect the different phases of these trials to verify that you are fulfilling the procedures that have been approved. Also, the quality assurance program may inspect or audits, which are the ones done specifically to the data, with the end to try to verify that the way that the data is being gathered has been done under the principles of good laboratory practices. So these audits are done generally for a final report, and also you will see that will be accompanied by a list of inspections and a sworn statement. Next. With respect to the inspections that I mentioned in the previous slide, we had 3 examples. Here I will show you the first, which are inspections relating to the study plan or protocol. And what we try to do in this case is to verify the fulfillment according to the good practices and everything and anything that has been written about the study plan and each one of the parts and the chapters, and ensure that these go according to the good practices. And we try to fulfill the principles. The quality assurance unit, once we sign this document, this protocol, what we do is to verify that we approve that the content goes according to good laboratory practices, basically that these principles are being fulfilled. But it does not verify that the quality assurance is approving the technical and scientific content of the study, because that is not a responsibility of the quality assurance program. In the case, for example, of Costa Rica, we have a checklist where we do an inspection of the protocol or study plan that in many cases has helped when we do this a little bit before the signatures, with the objective to verify that we are according to all the necessary requirements. And if we need to modify or observe on anything according to the study director, it is important for them to know that the quality assurance unit found some sort of observation with respect to how these plans are written. Also, we have data auditing in final reports. What we do here is to verify that within the data gathering in any part of the trial, from the field to the lab, what we try to do is we did this procedure according to what is said in the protocol and according to the approved procedures. In the case of auditing to the data in the final report, this is important to verify that the report is being written correctly and it is exact, containing anything referent to the fulfillment of the principles required by good laboratory practices, and that we present the information in a correct manner, chronologically speaking, with a internal coherence, and that the data is complete, that they are adjusted to the good laboratory practices, that they're dated, signed, that they are done at the correct time, that we fulfill what Dr. Joe initially mentioned, that everything is done at the time and that everything is evident. So the quality assurance inspector can perform the necessary reports. In the case of inspections, everyone has a different format. These are quite broad, and try to find every one of these important points in the inspections to see if they go according to what's expected and to apply this information to the faculty facility management team and the person that is receiving the inspection, which will be the primary field or lab researcher. Next. Very well, this slide is basically an example of the initial part of this checklist that we have in the quality insurance program. Each one of these programs will accommodate each one of these checklists according to their country. So what we do in Costa Rica is, this is the second page because the first page is description, basically the name of the study, the trial, the study director, the date, the site, and this will end up being basically like the second page. And each one of these inspections will have different extension or different length according to the necessary points that are being mentioned. And these points have to do with what the OECD. I have some background noise. Somebody maybe have an open microphone. Please be careful with that. If somebody could help me with an open mic we have, thank you. For example, here on the left, a small part of this checklist for the inspection, this would be the protocol and the study plan. And here we see basically if it's well identified, if it has the relative information with respect to the sponsor, the trial site, the dates, the methodology, and it gets quite extensive from here. In the case of the picture that I gave you on the right, would be an example of what this checklist would be for a final audit report. So here we see each one of the sections that are verified. It's quite extensive, as I mentioned, but here we see that everything was fulfilled according to the good laboratory practices, and we write down if they are here. If it's NA, then it's not applicable. Next, with the inspections that I mentioned 3 slides ago, this is another one of the inspections, which are inspections related to the facilities. I don't know if you remembered that I mentioned and visualized the facilities to be the physical building. It includes everything. So with these inspections relative to the facilities, the program of quality assurance tries to allow these facilities to be adequate to develop the studies according to good laboratory practices. So the quality assurance person will ensure that the lab or the places where the research is taking place, that these spaces are adequate, that the test substance is adequate, the storage of all the different sources, and the people working in the trials, that these are adequate people, that the number of people is according to the studies that are taking place. And this will happen from the field on forward in the lab and in the archive as well. Not only visually seeing this, but there are also things that must be verified. For example, curriculums or the profiles of the people working there. We need to visualize how these are being stored, as well as the files, how the buildings are, and to verify that all the different sites of the facilities will not interfere negatively in the execution of the study or the non-fulfillment of good practices. Next. An example of what we do with these inspections, or a form of these installations. Here we have the facilities, the substance, if they're well separated, how we manage the chemicals, how these are being used, how are we storing them, as well as controls from the lab, that they're properly labeled, the equipment. Just the same as I mentioned before, these are quite extensive. Next, please. And finally, according to the procedures, we have those that relate to inspections. As the name says, what we're trying to do is that to fulfill everything that is stated in the procedures that were previously approved. So what we try to do is that we fall upon the guidelines of the protocol and the procedures, that everything is working correctly, that the data is being registered correctly according to the good laboratory practices. And in the same way, calibration is done according to the good laboratory practices in each one of the procedures that have been written down and authorized. So with these inspections, we try to guarantee as a whole, throughout the development of the activities, everything that was written down in the study plan. And therefore, we have such an importance to a study plan that has been well revised and approved. But not only this, but that the procedures have been described and are complete for each one of the activities that are being developed by the investigator, are fruitful, and that the person in the quality assurance program may verify its fulfillment. It is very, very important that the quality assurance program verifies the dates of each one of these procedures to ensure that these are updated procedures. In the case of PPL, then it requires that the procedures are revised frequently, and also that data collection on the field and on the lab are done over the registries that have been previously approved by each one of these written procedures. Also, it is important that the quality assurance verifies that the data is being written, for example, in the field notebook at the time that they were taken, that these are as true as possible, that these are signed, dated, and that these are done with a black ink pen. The quality assurance verified that there are no blank spaces, that everything is done in the most transparent manner possible, that if afterwards we need to verify the trial, then these can be replicated because everything is well documented. Next. For the procedure inspection, then we do what we call analytical inspections on site, which is basically defining different phases or critical stages in which we described a procedure for each one of these phases, but also where we consider very important for the execution and the success of these studies. So within these critical phases, for example, on the field, when we do agricultural equipment calibration, when we do application of pesticides, and when we do fruit sampling. These are examples. Also, the reception and the storage of the samples. A critical stage may be the sample homogenization, another one, an analysis and quantification of the samples, and also the validation of the test method. So the quality assurance program must see that the quality assurance verify or inspect at least one phase of each one of these trials. So in this way, we may have validity over the quality, especially when the study directors may be far away or in another country. This may be very relevant, but because for them, it is more important or more evident that each one of these stages, critical stages, are being done correctly in the performance of the report. One of the recommendations that we do with respect to quality assurance is that, for example, if we have a study with 5 different trials, that the quality assurer ideally goes to the first sites in case that they detect that there's some sort of deviation or any action that might not be taking place correctly may be detected on time, and we do not compromise the work being done in the other sites. Next, please. An example, basically, of an analytical inspection on site. This is the second part. The first part describes the phase of inspection. For example, we inspect the third application of coffee plantation, and here we begin to throw down or go down a checklist with each one of the phases. In the case of Costa Rica, we have a very broad list for analytical inspection, which includes field and lab work. If I am working on the field, then the lab part, then just I write down NA, not applicable, in the same case, which is the different, or vice versa. When they work on the lab, then they put not applicable in the parts of the field work. I take advantage of this opportunity to say that these must be filled completely, and trying to fulfill all the good laboratory practices, writing down with black ink pen as clear as possible. The description is very important because sometimes the test facility management or the director are not on the field. So they must understand what is it that the inspector found. And in the case that there is not something that's not according to the standards, to be able to revise or to do any deviation. Next. In the case of quality assurance findings, there might be several types: serious deviations, minor deviations, or opportunities for improvement. In the case of serious deviations, those are the ones that, as the name indicates, are grave and jeopardize or seriously compromise the integrity of the study because of what is determined. Maybe, for example, it might be not fulfilling the GLP principles. The quality insurer must communicate this immediately to the test facility test management and the people necessary, the primary field researcher. And these are the people that will make the necessary decisions based on the descriptions of the deviations and the continuity of the trial. Also, there are minor deviations, which are the ones that don't really affect seriously or do not compromise the scientific integrity of the study or the ability to conduct these studies in accordance to the GLP principles. And then the third one is opportunities for improvement, which are where we estimate that it's convenient to make a change and that this is of interest to the GLP principles. It's not that having a recommendations of X or Y thing being said, or done because the opportunities will be over the fulfillment of the GLP principles. It's not something technical or scientific. Next, please. And as I finish my presentation, there's an important part of the quality assurance, a part of the auditing process. The final report is contemplated, which is an affidavit of the quality assurance program. And this is something that is required by the GLP principles for it to be included within the report. And that here in quality assurance, we have details upon which the study and where it took place, ensuring that it was performed under the best practices and that the final report that was revised after we included all the data, that as a whole, these are original data that are fulfilling good laboratory practices. According to the sworn statement or affidavit, we must perform a list, generally a checklist, of the nature of the inspections that took place, describe basically each one of the type of installations, critical phases that were inspected, the dates in which the inspections took place, and the dates upon which we communicated the results of each one of these inspections to the different people involved on the field or on the lab. It is important that this chart is very much thorough, and it is important, which I mentioned earlier, that the quality assurance almost always is paying attention to a received confirmation of the emails and the reports that each one of these people send to ensure that they have been informed and to complete the sworn affidavit at the end of the study. Next, please. Here we have an example chart of a statement of quality assurance affidavit. Basically, this is something that might seem small, but this is a very important stage where the insurer states that the data is the original data and that reflect completely and precisely that the obtained result during the execution of the study were collected or were written down under the good laboratory principles, and describes briefly as a chart the different inspection phase with the dates and the notification dates. In the case of the statement or affidavit, it always has the date and the signature of the person responsible, or those if there's more than one.
I think that with this, I have finished. And now next, please. Okay. I thank you all for your attention. And basically, the program for quality assurance has a broad chapter that can be read in case there's any doubt. And then the forms for registration are also available. The case of Costa Rica, we also have them adapted to our inspections, and we can share them with you. Thank you very much, Veronica Hidalgo from the Phytosanitary Service of Costa Rica for the presentation on quality assurance in lab and field. We have participants entering. We've surpassed 330 already, and we thank you very much for the interest you've shown for participating in these 2 days of workshop. For the new people who have entered, we are very grateful if you can kindly write down on the chat your name, your institution, and your country. That will be extremely valuable because we don't have the registration information. And with this information, you can facilitate in the chat. We can map and monitor who is visiting us in the workshops that we have now. And of course, we're in databases to continue in the future we can communicate on new events or providing information that the foundation can have on this matter. Thank you for being with us and for giving us your information in the chat. For the people who enter once again on the top of the Zoom platform, you can see an option that says view options. In this option, you can select the language in which you want to see the presentations.
Está que tenemos traducción simultánea en español,
Please
inglés.
remember we have Spanish, English, and French interpretation and the PowerPoint presentations. We also have them available in the 3 languages, but you have to select the language in which you want to listen and the language in which you wish to see the presentations in PowerPoint. Let's continue. We're doing very good on time. I think we have about 10 minutes in favor. So therefore, I am grateful with the presenters because you have done a very efficient use of time. We're going to go now to the next principle in terms of facilities, where the presenter is Edwin Barbosa from Agrosavia, Colombia. Edwin, please, if you may. Thank you, Eric, and good morning. Please confirm if you can hear me. We can hear you perfectly. Okay, now I'm going to open the camera. Great. Good morning, everyone. And in reality, it's a pleasure, and I'm very pleased that we can have so many participants in this event, which I think is extremely necessary for all of the countries that are participating in this type of project. And it is a contribution to our fields in each of our countries. My name is Edwin Samir Barbosa. I am an investigator from the Agricultural and Livestock investigation entity in Colombia, Agrosavia. I've been part of the studies as a study director for a project we just finished in 2016, and the MRL was established in 2018 for spinneret in avocado. And I was also working in 2 of the projects that we have with pineapple and another one in banana in Colombia. Today, I'm going to talk about one of the chapters of the GLPs around the facilities. Next, please. What are facilities? So facilities are basically, as Veronica said just recently, this is not only about the physical space, although I will talk a little bit about the physical space where we are developing this project. Basically, we have 2 types of large facilities in terms of the physical side. One of them is the lab, where we are accustomed generally to have different areas, and in the different labs, mostly those that have a pesticide residue work, we have areas where we have different activities carried out. For example, we have a standard reception area. We should have this area a bit secluded somewhat because we are receiving pure compounds, or with a high level of purity rather. And we also need to have the integrity of these compounds. Also, I think there's audio in the back. If you can please close the microphone. Thank you. Okay, we also have an area for sample preparation where we do the development and preparation of extracts for injections, and the instrumentation area where we have all the devices. Mostly in this type of study, we use chromatographic and liquid chromatography with mass detectors or with gas chromatography as well. We also have to have a location for storage or archiving the documents that are used in the lab. It's necessary, and we will talk a little bit about this briefly on these sites, but we have to consider that these places should be, to a certain extent, restricted for staff that is not from the study, but we also are looking for those who are participating in the study. So we have a first facility that's very important for the study, and that's the lab. And we'll talk about what that lab should have. Second, we have another large component, and those are the field facilities where we will develop the field trials. These field facilities are quite necessary to the extent that we have to select sites that have certain very special characteristics. We're going to talk a little bit about them, but to give you a little bit more information about this, we have to have places where there is sufficient area and where we can have a sample amount that's representative for the analyses that will be done in the lab. We'll talk a little bit about what these facilities should have, these field facilities should have. Also, at the field, we have certain archive files and sites where we need to store, for example, the substances for testing, this with completely restricted access. The field investigator is the one that is liable for these substances, these pesticides, having all of the characteristics, humidity, temperature. We have to guarantee the characteristics of these substances throughout the studies. And these conditions should be guaranteed, meaning we should be able to show with evidence under data logic records the temperature, since they should have a chain of custody until we finish with the tests. We're going to talk about this second large or significant component, which are field facilities. And it's also necessary to consider the sites in the field where we will store the application equipment that we use, as this should be very safe space. It should be clean. It should be spaces where there is no manipulation from the staff if these people are alien to the investigation, and where we can guarantee that the devices and equipment in general are in proper condition to perform the trials and the tests. Next, please. So within the requirements and best practice, good laboratory practices, I would like to highlight that more than meeting a standard for following these studies, what we should do have is a philosophy for good laboratory practices, so that we can feel at ease that everything that we're doing in the study is being done under certain scientific rigor guidelines, also having evidence of this happening. So any deviation that might occur within the protocol, which is the main guide of the studies, and also with the SOPs that we have for each of the activities, whether it be in the lab or at the field, we should understand that just as we wrote it, it should be done. And in case there is any deviation or that something cannot be met, as Ken said at some point, we have to inform the study director, because we have to take measures so that the scientific rigor of this is met. And the general information around the facilities, We have to have facilities with sufficient area to further the studies. And I'm talking about the lab and the field. In the lab, we have a lab that should meet with separate areas, as I said previously. And in the field, we should have enough area for us to be able to establish the lots that have been treated, the parcels that have not, as we do in the study. We should also consider that in the areas where there is manipulation of the pesticide or of the substance of reference, they should be completely separated from those who will not have that manipulation. One of the main, I'd say, rule of thumb for facilities is that they should be completely avoiding any contamination in the areas, both at the field and in the lab. So we should guarantee the integrity of the samples that will be taken both at the field and in the lab.
Es posible, por ejemplo, que en el campo
In the field, for example, we should be able to have, I think there's another microphone open right now.
Okay. Entonces, uno de los aspectos
So one of the aspects we should consider when we go do a study of this sort is that we should guarantee that the space where we will develop this should have enough area. Generally, the protocols, the study plans generally say that they should have twice as much of the sample that I need to take to do the analysis in the lab. However, we have to consider that in the field, we can have things such as any type of early maturity of any of the fruit, ripeness, sorry, of any of the fruit that we have there. And we have to consider this when we want to establish the size of the parcel. So there's fruit that will become ripe quicker than others, and there can be some that have some sort of phytosanitary issue that might be small, and they should be considered also for the harvest, time of harvest. And we also have to consider that, generally speaking, we don't take samples from the borders because the application in borders isn't very uniform if we compare it to the center of the parcel. So we have to consider each and every one of these sort of aspects so that the size of the parcels that are treated and non-treated in the studies should have enough
suficiente área y podamos.
area, and we should be able to meet this. Next, please.
Okay, siguiente, por favor.
Next, please.
Exacto. Este es un ejemplo, por ejemplo.
This is an example of the parcels that we use at the field where there is a parcel treated that is sufficiently separated from, sorry, a non-treated one that is in far enough from one that is treated. So at the field, we have to consider the crops that are next to these crops because they might have some type of contamination in our into our sample. And we also have to consider the separation of parcels that are not treated versus those that are treated. There should be enough space so that we can guarantee the integrity of these samples, that we didn't have in the cross-contamination, whether it be with a nearby location that was there or any other reason. So it's necessary that we consider this type of indication when we select a field facility. Next, please. In good laboratory practices, we have section 3.2 related to test systems. In this, we show that there should be enough area of separation between the test systems in the projects that use substances that can be organisms or also chemicals that can interfere in the sample contamination. In the case of the lab, we should totally separate the areas where we have the analytic standards or where we have other types of samples that can cause contamination on the samples that come from the field. And in the field, we have to be very much aware of these plots not having any contamination of applications that are near our crops or that are also with agrochemical storage that are very close to these treated plots that can contaminate the trials. A second point refers to diagnosis areas to prevent deterioration of our samples. It's necessary for us to consider that when we are at the field and we have an area for this trial, this area should be free of severe phytosanitary issues. And it should also consider that when we are running the different applications and we're going to take the sample at a certain given time, let's say 35, 42 days after we started the first application, it's necessary for us to consider that at that time we should have enough amount of sample that doesn't have any type of deterioration or damage so that there is a representative sample of our trial site and they should reach our lab in a healthy condition for analysis. We should also have storage areas, both for the different supplies and equipment.
separadas y como les comentaba anteriormente,
These areas should be separated, and as I said before, they should be fully marked and delimited as independent from our sample so that there is no cross-contamination. We have to consider that where we say reagent or reference material or the test substance where we have the different elements that we're using in the trials that should be clean. There should be a minimum maintenance and cross-contamination should be avoided. Next, please. What do we have to consider to have a proper trial site? First of all, this should be very representative. In our countries, we should select certain provinces, departments, or sites in the country where there are representative crops with the test system that we'll analyze. So let's say, for example, I'm going to have a study with pineapple. The ideal situation is to select sites in the country with representative pineapple crops with the same commercial practices or that consider the sale of pineapple both in nationally and internationally. These studies have to happen under commercial practices. This is usually how we do it and seeing how things are actually commercialized in real life. When perennial crops exist, that should be established already, meaning in the case of avocado, for example, or in the case of anything that is perennial, I don't have to be based on a small tree, but the crop should be established and it should be in a productive condition. I should establish my trials, and that's once the crop is close to being harvested. Because we're looking into the residue of pesticide in the fruit. So I don't have to wait until they're small. So for those who aren't perennial, we can establish these trials with from the point in which the crops are planted. These crops should continue with good agricultural practices in the area. This helps us have the registration that we can have weather matters, a history of pesticides that have been used, and also the practices of fertilization that we have in the crops. And in addition to having good practices that guarantee that the crop is maintaining in the best way possible throughout the trials. Places should be very easy, of a quick access, and the environmental conditions are very important. The environmental conditions as rainfall, temperature, and they should be registered. We should select a site that should meet this, and there should be weather reports that are very close to the crops, and they should be easily traceable. As I mentioned before, there should be we should have enough area and separated areas. We have to be cautious with cross contamination. Next, please. Here, we're going to talk a little bit about some of the characteristics that the plots will have at the field. First of all, we have to have a general direction of where these trials will happen. It's important, and mostly for collaborative trials where we have the 7, 8 trials that are necessary for a study to determine a maximum residue limit. We see this in different countries. This is important for us to have a location of the province, municipality, department, where the trials are happening based on the general map. This facilitates that the person that is looking into the records can be located in terms of space, where to know where these trials happen. This is very important for collaborative trials because sometimes we don't know the geography of other countries that are participating, and it would be very good that in these maps we can locate them easily where the study is developed. And it's necessary that from the closest population, there's a general direction for the field in the area where this happened, should have a description of how to reach this point geographically. It's important to have the distance of entry, for example, entry into a farm to the specific plots where we will be working on these trials. Mostly for places where we have trials in perennial crops. And this is when we have to be clear on how we can take necessary measures and what we see in the field book to see where the distances are from the entrance of the farm, showing where the northern direction is in the plots, to know where the source of irrigation is, if it's a lake, a crook, in some a creek, sorry, if we have maybe from the aqueduct, we have to specify where the source of irrigation is. We also have to know where the wind direction is. It's also very important because it can give us indications of what can happen with the application of pesticides. And as I mentioned, there should be a station, a station to measure meteorology, and we need to see the amount of rainfall, the temperature changes in the plots throughout the time in which we run the trials. Next, please. When we are planning these plots, we have to consider certain aspects, such as, first of all, we have to consider that the plots that we will apply these pesticides on, they should be located in a way in which the application of that pesticide through wind will not contaminate the untreated plot. So let's say it's very important to consider this because the wind direction is extremely important in terms of the derivation of these pesticides that we're applying. We have to consider these buffer areas that are, we have to consider the separation of these crops. We have to see these in the parcels, and we have to see an area of separation and the untreated one and the treated ones that we are in the trials. So they should be sufficiently separated. In some of the trials, it's 30 meters, but preferably 60. And we have an area where the crops are very, we should see this in the areas. We have to see the direction of the north, and we have to look into the slope of these plots. It's important that when we establish these plots in the field, we should consider that we are doing precision trials, not quantity trials. So what we have in these parcels, or we take out of these parcels, is quite accurate. So we have to select areas so where the person that's applying, they can walk and they can have a uniform application. And we have areas in, if possible, of a low slope, but we have to consider what slope there is in the site. Some places it's completely flat. There was a trial we did here in Pineapple, in Yopal, which is a city that is about an hour away from Bogota via plane. This farm was completely flat. But now we are doing a trial with the slope of the site. It's varying between 15-20%. So it's necessary that when we establish these plots, we shouldn't do this in very sloped areas because that will create issues with the trial. We have to consider the different numbers of lines and the direction that we have, the number of beds, the number of lines, rows, sorry, and the distance between plants and rows, establishing these plots. All of this is to be documented in the field document. Next.
Well, this is an example of what I mentioned, and this is part of the field notebook that maybe you have seen, where you can see the slope with some distances that we must take onto reference sites that are necessary. Generally, we have two distances to fixed points. Also, we must indicate the meteorological station, the distance from it, the distance between the plants, the distance between the rows, the area of the treated parcel, in this case TR0102, the necessary area, the distance between the treated and untreated parcels, which should be a significant distance, even up to 90 meters. And we also must indicate the slope of the terrain and the direction to the north. In the field notebook, we can see a very clear explanation with all the different factors that we must register as part of the data and setting up the field correctly. We must take all these factors into account. And piece of advice that I could give you from my experience is that also selecting these sites must be easy access, relatively close to airports or the place where the data analysis will take place. Generally, the protocol established that not more than 12 hours must pass between the time you take the fruit from the tree onto the freezer. So for this, you require to have good logistics to ensure that the samples will arrive in the necessary time frame from the field to the lab. In our case, where we have a quite broad geography and where we need to travel in planes, we need to take into account of the time the sample takes at the airport and how long it might take in the packing, in the delivery. And many of these factors must be taken into account. So a good piece of advice that I could give you is that these are easy access areas, areas where we could possibly be close to sites where they can be shipped to the lab, or they are close to the lab in some way to accelerate that logistical time frame and to be able to fulfill the necessary requirements of the protocol. Next, please. Very well. In the lab, and as I mentioned on the field as well, we need to take into account that the reference materials, we're talking about analytical standards that come certified under these commercial businesses that have gone through a certification process and have pesticide or metabolites with respect to toxicology, and also those isotopically framed. These materials must be kept in an area where it ensures the storage and temperature, where we have specific registries of their use and also their integrity. The temperature must be monitored from the time they are received to the time that the trial ends. And we need to ensure in this time frame that if we're saying that the study took place during 2-3 years, we need to ensure during that time the environmental conditions where these chemicals must be stored. It is necessary to take this into account. And also, this is very clear for the labs in areas where these samples or the sample treatment is, because there cannot be cross-contamination. We in the field must have storage areas for these pesticides, and these must be completely separated in some case. We want to store these systems. These must be completely separated from the samples. In the case of the lab, when we receive the samples from the field, these samples must be stored in a cold room or a freezer, completely separate from other things that might cause cross-contamination to the sample and the reference materials that we have. It is very much important to take into account that these labs must separate the untreated samples from the treated samples within the drawers or the different spaces in the freezer. There must be a physical separation between the samples, even though the samples might be packed inside of plastic. So there is no direct contact from one sample to the next. Also in the freezers, they must be completely separate. So it's important to take into account these good laboratory practices to have one sample next to the other in the same freezer. So it's necessary to keep them separate. Also, it is necessary to maintain the integrity. All the samples must be well labeled, and this labeling must correspond to what the protocol says or what the plan, where it states the different activities of each one of these activities that are being developed both in the lab and on the field. So here we must have a total identity that fulfills the requirements of the protocol, and also it is necessary to take into account that there must be a registry in the laboratory, and this is important to take into account. There must be a registry of when I take those samples out and in of the freezer, And this is necessary because there might be a traceability that the sample has not lost its chain or its temperature control, and at what time the sample left the sample temperature at which it must be kept. So it's important to have a registration of when the sample came in at the first place and when it was taken out of the freezer to go through the internal processing of the lab. Next, please. Next is this part of the talk has to do with the archive facilities. It's important to take into account these archives. Let's say that we'll have them throughout the trial, and at the end, we'll have some bigger files which correspond to the lab and what corresponds to the field. All these parts of the general files must lay within a site where they are protected, where there's no deterioration. General good practices include that these archives are placed physically in paper. So we must prevent the deterioration of them to get wet, damp, or water temperatures may be leading to paper deterioration or whatever might happen to those files. So they must be stored in a location that is very well protected of any conditions that might harm them. In these files, where at the end of the study, we have all the different files, all the different audits that we have, and everything that we've done doing on the field. These files must have limited access. Not anyone can go and use this material at any time, and it should have an authorization from the study director to avoid these being lost, or in the worst case scenario, for them to be modified. And they also must be organized in such a way where you can have easy access. One of the things that we managed to do, for example, in the avocado program, which was our first experience with this program, is that in completely well labeled boxes, we have the files that come from the field, and in another box, the quality files, and another box, lab files. And in each one of these boxes, we indicated who placed what document in that box and the state that they were in. And also there's a use control. So, for example, for these trials that we're doing now, sometimes we need to go back to previous studies and see how we did that 2 or 3 years ago. So it's necessary to be able to have access to any of these documents for them to be a use control. And generally, the director of the study may give the authorization for that document to be revised and given back to the file as quick as possible. Within the lab, when we're working on lab trials, it's necessary for these archive files, whether it be chromatograms or the formats used for the use of mixes, calibrating the equipment, all the document bunch need to be separate from the area where we're developing the sample analysis to avoid any type of contamination or deterioration of the files. This is what I wanted to mention with respect to the facilities. It is very important that for the processing assays, those that need to go through these procedures, it is important for us to guarantee there will be no sample contamination. This cannot be done on the field. We need to have very well isolated sites where there's no cross-contamination, where we can guarantee the temperature of the sample totally adequate for that type of processing. So in many cases, it's sometimes easier to do this directly on the lab to avoid any type of this contamination. And the last slide talks about waste. And we're talking about waste both on the field and in the lab. In the field, we need to have a system to get rid of these standards that are well established that is generally do in a lab. This is very practical for people that already have implemented 17025, but the ones that don't need to have a registry where we see a flow in the lab that guarantees that this waste will not come into contact with the sample or with any type of element that is part of the trial. And also, we need to define procedures for the transportation of these samples and the elimination of said samples. In the lab, in the many of the labs that are already part of going through getting certified, this is very easy. It's a requirement, not easy, but it's a requirement that we have as a set of labs. And also on the field, we need to take into account that these samples that are being thrown away in the case, especially where I have my treated parcel, I take my samples, and as a matter of fact, I send my samples to the lab. I ensure that the lab receives the sample and that I am I'm finishing the trial, so I'm gathering all the fruits that were treated in cases such as ours, where the pesticides are not registered in our country. It's necessary to take these and to bury them to avoid these fruits contaminating any from the commercial crop, or that they may be commercialized by the farmer, which would be fatal if they're not registered in the country. So it's important to take this into account, and also to take into account that when the crop, at the time of the crop, we always must harvest the untreated parcels and then afterwards the treated ones, making sure that from every sample that we're taking, avoiding the edges of these parcels, and possibly broadly that the sample, once it is taken, goes directly to the wrapping, so there's no contamination with the ground or with any type of element that might contaminate our sample. Obviously, that the person taking the sample is using the appropriate clothing or equipment, and that they change their clothing so there's no cross-contamination. This is what I wanted to mention, and I go back to you.
Muchas gracias, Edwin.
Thank you, Edwin, for this thorough presentation about installations or facilities. I have seen that some of the participants have been raising their hands on some occasions, and I remind you all that after the presentations, there is no time, or we have not enabled time for Q&A. So please write your questions down on chat, so Veronica can be gathering all these questions, and they may be answered afterwards at the end of the presentations in the section that we have dedicated exclusively to the matter of questions and comments. Let's go and let's continue. We have two more presentations, two more principles, and the next has to do with materials, teams, and chemicals, in charge of Brenda from Panama. Brenda, thank you very much for your presence here and for offering this presentation. The floor is yours. Thank you. Thank you very much. Thank you for the opportunity. My name is Brenda Checa. I am a chemist, and I work in the Ministry of Agricultural Development in Panama, and I'm responsible of the quality, pesticide, fertilizer, and residue lab, and we have been working on these projects for the establishment of maximum limits. And we began with the residue of Proxifam and pineapple. Now we're working as a team, as in research investigations in Panama and ourselves, with some cooperation and the minor use foundation in pineapple and banana. And it corresponds to me to present to you all, speaking about the apparatus, materials, and reagents. Next, please. The apparatus and fundamentally the informated computer systems used for the generation, storage, and the retrieval of the data of the controlling environmental factors relevant to the study should suitably be located and be of appropriate design to this capacity and have enough of all the computerized system used to produce or evaluate data with normative aspects should be validated, operated, and work within the product of good practices. Next, please. For each application or system, we must implement logical measures to avoid unauthorized access to the information system, applications, and the data. It is essential to ensure that only we use appropriated versions and validated software. Logical security may include the need to enter a unique user, identify a sole password associated, and we must control any type of data introduction or software from external sources. These controls may be given by the operating system control of the computer, specific security routines, and those integrated in our systems or any combination of the above. There is an application of good practices specifically to those information systems that are part of document 10 of OECD. And here we can revise here from our point of view, working on a lab and all those that use Excel spreadsheets and good lab practices and other norms. It is very important that when we make use of these spreadsheets, these must be validated, codified, and blocked, so the formulas cannot be modified. Next, please. The field apparatus used in the study should be inspected, cleaned, maintained, and calibrated in accordance with the normalized procedures. We must have registries of these type of activities, the calibration, and if they would take place, to store these according to the national standards or even international standards. For example, the matter of thermometers, where we can assign an ID number with a sheet or a log where it registers the temperature, the calibrations, and all the verifications that have taken place, any harms, any damages, repairs, any information from thermometers or any type of equipment that we're using is important to be able to identify these. The next, please. Field apparatus and materials used in a study should not interfere in a harmful way to the systems. And it's important to be able to ensure that this takes place. For example, in the picture, we see within the materials to use on the field, it is important to correctly label the perimeter of these parcels with a warning tape to find it easily, Also to label the plants in the area of study, where we will select an area for the trial, also to make use of these materials that you see here and the metric tape to be able to have any necessary measurements on site is important for this type of work. Next, please. Also, within the site on the field for the applications, we will need basically water pumps for the pesticide, a toolkit. It's important to have any repairs, any parts, repairs that might be needed on site. And it's important to have a psychrometer to measure relative humidity, to use an anemometer to measure time or wind, excuse me, and the temperature of the wind. Also, we have soil thermometers to have a depth, maybe 5 or 10 centimeters under the soil, according to what the protocol mentions. In the case that it's a rainy season, then we must have also a way to measure rainfall. And it's important to take into account that a chronometer might be useful in the time of the application. When we apply the pesticide, it's important to measure the time during the time of application. Next, please. It is very important to prevent during the time of the study applications where we might see any type of contamination of the equipment that we might be using. It's important to have all the necessary materials to be able to perform the mixtures, in this case, of the pesticide that's being studied, and also to have the necessary labels. Here we're working with, in the case of bananas, here will we put the site number, the site location in the study at that time, also to have containers, maybe 20 liters to perform the appropriate mixture. Also, when we are going to measure the volume of pesticide that we will apply, to have the graduated cylinders, containers that are required at the time, as maybe a syringe. Here we have on a picture an application of spinetoram and banana, and the person applying this is the person on the right on your screen, and then the person on the left, and the picture is taking time. Next, please. It is important also within the materials that we use in the data collection process that we prevent contamination of the samples that we're taking, specifically in the case of trial substances that might cross-contaminate at any type of time after the data sample collection. Let's ensure that the materials are at the appropriate size, clean, and that the bags or the materials must be made in a material that don't affect adversely the associated analysis or that absorb residue. Any type of device such as a knife must be cleaned thoroughly with water, soap, and a brush to eliminate any type of contamination. Finally, we get contamination if we don't use ammonia in many of these cases. And for the data sampling or sample collection, we will need gloves and some bags that in this case are fabric with plastic inside, and touching these samples with our hands or clothes that might be in contact. Let's avoid this. Let's try not to use or have the try not to contaminate the samples that we're using, and at the same time to avoid contamination of other type of food sources. Ensure that these are not in contact with equipment that have been used before to applied or to transport any other type of pesticide. And for the data collection, also in the case that we see in the right, we will need a ladder in the data collection of these bananas. Also, we will need to make use of disposable boots. The next, please. It's important to take any necessary preventions for the transportation of the samples, and therefore we will need to have a gel pack or dry ice if we're going to transport these samples, as Dr. Barbosa mentioned, that we must get these samples within 12 hours to the lab. And that's why they must maintain at the temperature that the protocol states, and to always be equipped with a cooler gel packs in this case. Also, it is important that to measure temperature, we must be sure of the temperature that the sample has had during its travel with a minimum and maximum. Next, please. In the case of the apparatus, the chemicals, these must be completely and thoroughly labeled with a concentration, if that applies, the expiration date specific to its storage. It is necessary to have information with respect to the origin, the date of preparation, and the stability. The expiry date may be extended based on an analysis, based on a document evaluation. And when we work with these samples in the lab, any type of equipment and samples that we're using are not contaminated, that they are well calibrated, that we maintain a log of all the information that we might need, so that the results are reliable and precise when it's time to perform or write the report. And this is my presentation. It's quite short. My principle was very short. Thank you all very much for your time.
Muchas gracias, Brenda.
Thank you very much, Brenda Delmida from Panama, for this presentation. Fortunately, it was a short presentation, and we have very much set ourselves on time. We are reaching the end of our presentations. We just have one more. The next principle and test system, by Mr. Edwin Barbosa from Agrosavia, Colombia. Without further ado, Edwin, you have the floor. Please go ahead. Thank you. Thank you very much, Eric. I think you can all hear me. Is that correct? We can. Okay. So in the last presentation, I spoke a lot. So the reward is that this one will be a little bit shorter. What is a test system? Let's go to the next slide, please. I'm going to talk a little bit about what test systems are. So according to the GLPs, the test system is designates the totality of biological, chemical, or physical systems, or a combination of the above used in a study. In our case, we can have test systems with animals, plants, bacteria, organs, cells. But in our case in particular, for the maximum residue limit and establishing them, our main test system will be the species where we're working, whether it be pineapple, cocoa. This in terms of the vegetable material, but we also have other ingredients in these test systems that are all of the systems of chemicals or physical systems that we're using for the study. Next, please. According to the GLPs, all of plant seeds or soil should be described broadly from the source, its acquisition, variety, and any other feature that identifies it. In our case, in the test systems where we use species such as pineapple, for example, we should identify the variety of these pineapples that we're using, where it's located, where we take the samples from, and all the information we can have around establishing these crops. The same happens for any other product of the agricultural products we're working on. In maximum residue limits, it's necessary for us to have a characterization of the soil where we have established our crops. However, generally speaking, these soil studies generally are beyond the GLPs. However, since they're not part the test system is the species where we're working on. However, this soil, although it very well describes how it could be comprised, it's usually beyond the GLPs, but it's necessary to have them. So all of these studies to meet the GLPs in the different countries and with a soil analysis contained by the farm. The test system says or states it's the species we're working on, and we also need information from the surface, from the ground, that shouldn't necessarily be under GLPs, but it should give very reliable results regardless. For the case of test systems for pesticide studies, they should have very special characteristics. Let's go to the previous one. They should have very special characteristics. When we are at the field and we're going to certify, it's necessary for us that we consider that I cannot do an application of the product when the plant has any type of dew on it, or if it's still moist after raining. It's necessary to consider that these things can cause external interference from the test system, and we should wait for the dew to dry or the plant to dry to then apply the product. Generally speaking, in the study plan, they discuss a preliminary analysis or pre-treatment of control samples. They are described clearly to see how they should be done, how we should do this treatment of the samples. It's necessary that we continue and following into detail what the protocol says in these test systems. In case there is any requirement that cannot be met, we necessarily have to report this to the study director. And if there's any deviation, tomorrow we'll see what a deviation is and what an amendment is. If there is any deviation, meaning anything that was not done properly, and this with respect to the protocol, and we have to report this to the study director to take the necessary measures. This we have talked about, this minimizing contamination and interferences. Next, please. For the case of the crop registration, then in the field book, we have this in page 16. What we're working on, the variety that we are working on. Generally, in the studies, in the 7, 8 trials that are part of the study, they analyze different varieties. In the case of pineapple that we're doing currently, in Colombia, we are analyzing a variety that's called Gold, MD2, Gold Honey, Oro Miel. And in collaborative trials we've had in Bolivia, mainly in Bolivia, because I think Ecuador is also M2, we had a variety that's called Cucar. So as part of a study, there can be different varieties. The important point is to register what is being worked on in the trial site and the methods. If this is through seeds, if this is through plants, transplanted plants, or if the crop has been established in the case of perennial crops that I mentioned, this is also part of the registration done in the fields. The age is also important, and the height of a vegetative state where the plant is also very important. For tropical countries, we have, generally speaking, we have 2 flowerings in different plants. We should register when the trial is happening, because this provides information in terms of this with the people from JMRP who are working on the status of the crop and when it was done. Next, please. As I mentioned before, in these test systems, this is not only with the species that we're working on, but also the physical or chemical systems or biological that are part of this study. In this case, the devices that provide chemical data is also part of these trials. And as we know, these should be validated. We should be able to prove this with support, with reports. This in terms of the validations of these devices and equipment to make sure that the results or the result that is being contributed is quite reliable. These devices, we have to identify them under a serial number that has a date where we received the equipment and registering the dates where we're using the equipment. Now, with software systems, it's easy in the sense that all the data comes with these dates when they're set out in the equipment, but it's necessary to guarantee that, in effect, we can have the registration. For the case of reagents and chemical products that are used throughout the analysis, it's necessary for these solutions to be well identified, having a date of preparation, an expiration date as well, and an instruction of specific storage. This to make sure that the different solutions or reagents that are used throughout processing these lab samples or obtaining these extracts in the lab for instrumental analysis can be proper, and we are not using reagents or anything that has expired, or reagents or combinations that have expired that can impact the result that we want to obtain. Now, in terms of We should have stability and storage in the samples that we received in the lab. And as I mentioned before, these freezers where we keep samples should keep a checkup of the constant temperature throughout all of the time that they have this in. This for the case of extracts. In some occasions, what happens in labs, they're processing to obtain an extract and to inject it in the device, and something happens throughout the procedure. And the device was, it lost its calibration, and we already have the extracts. And mostly it's 14 days. We don't allow more than 14 days of storage for these extracts, and this storage should be registered under specific temperature conditions. So we should guarantee that the extract didn't have any type of issue throughout the time we obtained it until we were able to analyze this. For the plants, it's necessary to maintain the integrity, that there is no degradation. For our case, it's easy in the sense that if we have a very strict cold chain, we won't have this refrigeration chain, we won't have this issue. It's not recommended, and it would be a severe deviation from GLPs that the samples thaw. So it's necessary to consider that one of the critical points once the sample is in the lab is that you should make sure of their refrigeration chain until it will be analyzed, and also after that, because we have to look into the results of residues. It's not good to thaw the samples. It's not allowed, actually. So the refrigeration chain should be something quite strict that we should have and consider. Now, in terms of bags and containers that samples have, the containers where we have the samples, when we have pineapples or we receive it, we look into it, we cut it, we keep the pineapple in the freezer, and then the next day we'll process it with dry ice to obtain a dust, and that's what we'll analyze. It's necessary for these containers where we're keeping the samples are new or are completely disinfected. They have to make sure that there is no contamination. So it's necessary for us to have in each step a consideration. So what I want you to keep from my presentation, that one of the critical points in this type of trial is a cross-contamination. We have to be cautious with that. We have to consider this. These are two images where they helped us. We have Dr. Amy Wine, and Milena were also helping us with this training in avocado, and they were telling us how we should peel the avocados to avoid cross-contamination from the skin to the pulp, because we were looking into processing. And this is an image you can see on the right of how we have to do this. So with all of the safety elements to avoid contamination. And in the case of the boards, you see there the knives between sample and sample, these should be washed, disinfected to avoid cross-contamination between samples. And the same would happen if you look in the bottom of these boards. The counter is also covered in plastic, and we change it between samples to avoid cross-contamination as well. And when the samples reach the lab with their label, they have their label, as you can see, completely packaged with a temperature sensor to secure the temperature since they reached the fridge and then arriving to the lab and then going into the freezer there. Next, please. The time in which the sample arrives to the lab, it should be identified with all the data, with a code. Each sample has the trial number and it's got an ID that the protocol states. Generally, samples A and B are non-treated, and C and then from then on are treated. So we should follow this identification that the protocol states. So the identification of these systems should at all times come with the sample. As I said before, if the sample would be analyzed, we should be fully clear on what sample this came from, when this happened, and when they go back to the freezer. And during the analysis, this is normal in labs. We should have a complete identification throughout the analysis process, meaning a single identification per sample so that there aren't possible maybe that the samples are changed at the time of the analysis. So there should be a single ID. All of the areas and containers should be vented and also disinfected, and of course divided in any type of interference. Next, please. This is what I wanted to mention about the test systems. And as I said, many of these things we will see in detail in the different principles of GLPs. I don't want to move forward in this topic right now. So Eric, you have the floor. Thank you very much for the time you've given me. Thank you, Edwin, once again for this excellent presentation. We have used time very properly, very efficiently, and I think we are reaching a very good time. Many of you have asked throughout the chat. Veronica Picado, consultant from the foundation, is responsible of managing this section, this Q&A section, and she has processed the questions throughout the workshop to try and give an answer to all of the questions or comments and what time allows. So Veronica, you have the floor to start with this section, please. Hello, everyone. I hope you can hear me well. We have 10 minutes more than what we expected to go into the Q&A section. So let's answer up until time allows. And if there is any question that we haven't answered, we can answer them afterwards when we look into the chat via email so that the person doesn't have this question unanswered. Lorena Lorca from Chile has the first question. Maybe if could you answer this, because I think we have this in the meeting. And they're asking if a same person can have several roles or responsibilities in a study. If it could be a study director or main investigator, when can this be done and when can it not?
Yes, it is possible for, say, a study director to have an active role in a field trial, for example, or in the lab, or for especially if there is a local coordinator named in the protocol, the local coordinator may actually participate in the study conduct. That is not usually how responsibilities are divided. I should say that the responsibilities normally are divided so that the study director is someone different from the field investigator and someone else is the laboratory investigator. But in some circumstances, it is okay as long as that one person is actually taking care of all of the responsibilities for that position.
Gracias, Ken.
Thank you, Ken. Here we have another question. It says, how many years of experience should someone have to be able to become a study director? And if they have to have experience on the field, in the lab, or in both, or how should that experience be built?
I guess that's for me again. I can't say there is a specific number of years. What is most important is that the person hired to be a study director receives good training right from the start. I was hired many years ago by the IR4 project to be a study director when I had been working in the field for 5 years. And some of that work involved residue field trials. But most of my training to be a study director was received at IR4 in my first few months, and then through the years I received continued training. So that is, and I had never worked as a chemist, and so I was then, and to this day, I rely on the expertise of the other people in the studies that I am involved with. I even after nearly 30 years as a study director, I still do not know everything. I need to have good communication with the other study participants, and that allows me to make good decisions for the ongoing studies. So I think if you are in an organization that is looking to hire study directors, you should be looking for people with scientific backgrounds. It could be, it's actually good if you can have more than one study director, to have one chemist and one or more field people, to have a mix of experiences. And they should be people who did well on their previous job and were responsible people. And if they had been working in good laboratory practices, had shown that they respected GLPs and carried out their work under GLPs.
Bueno, continuamos con las preguntas. Tenemos otra que creo que puede contestar cualquiera de.
We have more questions that any of the presenters can answer. It says, where can we find the best practice, the good laboratory principles? Where is that standard reference where people can seek this? They should see what they should meet in the lab. Where can they find these GLPs?
The OECD GLPs are available online. And I don't have the link handy, but if you look up OECD good laboratory practices, you should be able to find them. And they date back to, I believe, 1997 or thereabouts. Yes, 1997. And I had no trouble finding them myself when I started with the Minor Use Foundation. And so I think anyone in this meeting should also be able to find them. It's called OECD Series on Principles of Good Laboratory Practice and Compliance Monitoring.
Me parece
Ken,
que Joe también.
Yeah, Thank you, Veronica. Ken, I was going to jump in and say that, of course, they can find it, just as you said, but also Veronica or I or you can send a link in the chat or later on.
Correcto, para ampliar la respuesta de Joe al final.
To extend your or enhance your response, Ken and Joe, at the end of the event, we will be sharing a PDF, most likely, with the links of the main OECD documents where you can find the principles and for each of the principles, also how to meet them. There can be another specific document for quality assurance, and this for field test, and we can share the links for you. So we'll be sending you this at the end of the event.
Okay, continuamos con.
Let's continue with the questions. This question is from Uruguay, and it says the different stakeholders in the study protocol can have different sources in terms of experience. If these authors, they can come from official, public, private sources separately. They can be also from university sources. Joe and Ken, maybe if you can let us know. In addition, who should prepare the protocol? And could you tell us who can be part of the study staff? Somebody has the microphone open. If the study staff can be comprised by government entities exclusively, or can it be mixed between universities, government, focused especially on the trials that we are carrying out?
I'm not sure I understood the first part of the question, so I will start with the question about the protocol. Often it is the study director or the person who is intended to be the study director who will prepare a draft protocol in consultation with the pesticide registrant, because we have to be sure that the instructions in the protocol will match the instructions that the registrant is willing to support on the label of that pesticide. But anyone could prepare a draft protocol. And again, I would recommend that whoever writes the protocol is in contact with the registrant and also has information from the growers of the commodity where you'll be doing the study, to have some feedback on whether the way that the use instructions are being written will benefit the growers. You need to know not only from the registrant, but also from the growers, whether this proposed use of the pesticide makes sense. Ultimately, it is the study director who must sign the protocol, but the protocol may have been prepared by someone else working with information from registrants and from growers. I was going to answer the last part and now I've already forgotten. So if Veronica, if you could repeat to me the other parts of the question, I'd appreciate it.
Okay, creo que la doctora Laura Lombardo.
Well, I think Dr. Laura Lombardo from Uruguay, if we're not interpreting your question well, you can take the microphone and do it yourself. But I think what you want to say is that the staff from the studies must be from the same organization, or they are interactive. For example, the lab is done in a university, the field work is done by any type of farmers association. Is that correct?
Yes. It is up to the sponsor of the study, not the study director, but the sponsor, to oversee the selection of who will conduct. which organization will conduct field trials and which organization will do lab work. And then it is up to the facility management at those organizations to actually determine the individuals who should be named as the research investigators. But it is entirely normal for field trials to be assigned to one organization and laboratory work assigned to another organization. There are contract research laboratories that are not affiliated with any government or university that are often used in these magnitude of residue studies, especially for the laboratory work, but even for running field trials. And so that any one study may have a mix of organizations involved in the different phases and in the different trials within each phase of the study. The important thing is that everyone be trained in GLPs and be competent to carry out the instructions for their part of the study.
Gracias, Ken. Y vamos a dejar descansar a Ken.
Thank you, Ken. We will let Ken get a rest and we'll go with Edwin. The next questions are for Edwin. We have three different people, but these are related. So Edwin, I will do these questions back to back, and then if you need me to repeat them, I will. Carla, Areta, Diana Lorca, and Diana Lorco, I think about the storage of the samples in the freezer. The first question says if we can then use the same freezer to store treated and untreated samples. Lorena is more of a comment, and Diana is saying at what temperature should we keep these samples? Can you hear me? Yes, we can hear you. Go ahead. Well, with respect to the first part, with respect to the storage of these samples, what's ideal is that we have separate freezers to store the treated and untreated samples. That would be ideal. Nonetheless, in some labs, it is hard to be able to have two different freezers because there are other types of samples being stored or that require storage, and then we do not have any more freezer space. So what we need to take into account in these cases when we analyze these samples in the same freezer, first, that the treated samples must be in the topmost, should not be in the topmost part, because in case there is some kind of unfreeze or something happens with the freezer, most likely this will drop down and contaminate the other samples. Two, they must be in spaces. For example, if we have a 2-compartment freezer, it's better if one part is treated in one compartment and the other, and have a physical separation within the two. In some cases, one of the trials that we performed, where it was very hard to find a -20-degree freezer, it is necessary to have that separation. At the time, we did it with the separation of cardboard, even to separate them, and as far away as possible one from the other. What's ideal is to have, of course, 2 separate freezers. That's ideal. But if we don't, then we should have at least a logical flow for the treated not to contaminate the untreated. That's with respect to the storage question. And the second one, with respect to the temperature, the samples must be kept from the time that the samples are taken on the field. From that point on forward, we need to start to refrigerate them, whether it's using refrigerant gels or any other type of system such as dry ice, any system that does not interfere with the sample but that lowers the temperature, and with the data logger or data control that's annexed to each one of these samples that goes on to the lab. When they reach the lab and they have the initial processing, whatever it might be, before going on to the freezer, once they are in the freezer, they must be stored between -18 and -20 degrees centigrade. We must ensure this storage with the completely frozen samples. And we have shown that for these pesticide residues, these are ideal temperatures to be able to ensure that there's no degradation of the pesticide during the time of storage. These should not be refrigerated. They should be frozen to -18 to -20 degrees C. What's preferable is -18 to -20.
Gracias, Edwin. Joe, adelante.
Thank you, Edwin. Joe?
Yes. I just wanted to add everything Edwin said is correct. The separation of the treated and untreated samples is imperative in case there's a thaw and there's contamination. But I also wanted to add that another way you could ensure separation if you don't have separate freezers is for the plastic lined cloth bags, it's permissible to put each one of those cloth bags in a plastic bag and seal it. So each sample is in a cloth bag within a plastic bag. In that way, if there is some kind of thawing, that they won't get contaminated. The plastic should be adequate separation. Thank you.
Gracias, Joe.
Thank you, Joe. We have a lot of people in the audience, and possibly many of them, it's their first time they hear a bit about good lab practices. So I would just like to clear up to those people here that this is a quality control system that is generally applied to give traceability to studies where we represent some type of regulator to establish a registry or to establish, such as in our case, what the foundation does with each one of our speakers, and that's establishing maximum residue limits. So many of the responses are focused towards the residue trials and vegetables to be able to establish tolerance level. For example, with respect to sample temperature, such as indicated by Edwin, that is the case for pesticide residues in vegetables, which is mentioned by the regulators. But if you're going to perform trials in other areas, for example, the area of cosmetics, or any area that also applies, that you consult what would apply for this type of differential product. And we will continue with the questions. I see a raised hand. Yes. If you still want to ask that, Leoncio. Yes, Leoncio Iman from La Molina University. My question is, if we have a sample, we collect a sample on the field, what time can pass before that sample reaches the lab, both for physical and chemical analysis? Does it suffer some type of deterioration, or does the analysis suffers from that time frame? Thank you.
I will try to answer that. It is a complicated answer. The important thing is to get the sample frozen as soon as possible when it is collected out of the field. If the sample does not need to be cut, if it is small fruit and it is left intact, and if you can get the samples into a cooler with ice or artificial blue ice, then you may have, it is okay to have a few hours until you get it into a freezer. If you need to cut these samples because the protocol instructs you to do so, either to remove stones from the fruit or to separate peel and pulp, then it is very important for it to be cut, for it to be frozen very shortly after the cutting, because it is known that cutting fruits causes more rapid degradation of the residues than when the fruit are left intact. And that is why in the studies sponsored by the foundation, we recommend bringing the fruit to the laboratory in a cooler on the same day as harvest from the field, and then doing all of the cutting at the laboratory so that it can immediately be put into a freezer at the lab. But we know that that is not always going to be possible in all locations. So different plans may be needed for different locations. But I would say in general, cut samples need to be frozen within 1 hour. Uncut samples should be frozen within 6 hours, the same day, but they need to be in a cooler very shortly after collection from the field to help reduce any degradation before they are put in the freezer.
Gracias, Ken.
Thank you, Ken. We have another question here about the certifications under best practices, and basically is who must certify an analytical standard to say that it is certified under good lab practices? If we would need to do that under an ISO 17034, or how does this certification work, where the certified must have an analytical standard?
I will try to answer that one as well. Normally, it is the registrant who supplies the standard who takes care of the certification. It will come from a batch or a lot of the chemical that has been prepared specifically to use in GLP studies. And so they will certify that batch and then send subsamples to the various laboratories that need to have the standards. And then the registrant or the lab that they have contracted with to perform the certification will then supply to the study director and to the labs that receive the standards the certificate of analysis that confirms the certification. And the same is true of the test substances that are used in the field trials. They also have to be certified, and it is usually the registrant who makes sure that that occurs. In rare instances, there may be studies conducted with an older pesticide that does not have a registrant that is willing to spend the money on certification. And in that case, it would be up to the study sponsor to follow through and see that a certification occurs. But nearly all of the studies, maybe all of them, conducted or sponsored by the Minor Use Foundation involve test substances and reference substances that the registrant is certifying.
Bueno, continuamos con las preguntas.
Very well, we continue with our round of questions. There's curiosity to know if the variety of crops influence the result of the residue analysis and the result of the trials when these take place.
Oh, Edwin, do you want to answer this?
Bueno, Ken, gracias. Yo respondo y tú me
Very
ayudas.
well, Ken. Thank you. You can add on to anything if you want. Within a study, basically, we have different trials. And the idea of the complete study of these 6 or 7 or 8 trials that we need to present, the idea is that we contemplate a variety of things that might happen during the different trials, that these are taken in different conditions in agroecological terms, and that's why it's possible to perform these in different sites in different countries. But also this happens because we establish the LMR and the crop. There is a tolerance of the pesticide in the crop. In the case of pineapple, it doesn't say the variety of pineapple, it just says pesticide for pineapple. So the idea is that these different varieties, which are part of that variability within the trials, what's important is that these varieties really are a sample of commercial varieties of these type of crop. I don't know if Ken would like to add something else.
I agree with everything that Edwin just said. I will add that the number of trials needed for a particular crop might only be 4 or 5, and there may be many more varieties available. So we cannot include every commercial variety. So it is good to choose among those which are most often planted for the crop in that location. But if there are varietal differences which seem like they might make a big difference in residues, maybe one variety is very large and the other is very small, or one variety has smooth surface and another variety has hairy surface. That is an important difference which could produce residue differences, and that should be considered when choosing varieties for a study.
Gracias, Ken. Entró una pregunta que está en
Thank you,
inglés.
Ken. We have a question in English, so I'll pass on the microphone to Adriana so she can read that question.
Thank you, Veronica. We got a question from Uganda, and it says, how do you address varying crop quantities in the field during a decline study? For Ken, I think.
How do we deal with varietal differences?
No, Ken, sorry. With varying crop quantities.
Varying crop quantities in a decline.
Mm-hmm.
Meaning that perhaps the plot is not producing so much at the end of the collection. It is not. There is a minimum sample size that is indicated in the protocol for each sample collection, and that is important to meet or to come close to, at least if it cannot precisely be met, because those standards are set by codex, and they could potentially not accept the results of a trial if these sample sizes are not enough. But the most important sample is the one that matches what the proposed label will be, and that is you can tell if a study has 4 trials and only one of them is a decline, the other 3 will have the collection date for the proposed label, and the decline will have perhaps 5 different collection dates. So it is most important to get the correct sample size on that targeted pre-harvest interval. If necessary, if there are 5 sample dates and you're on your last couple of collection dates, you are running out of the fruits or beans or whatever it is. For decline data, it is really only necessary to have 1 sample, not 2. And so you could contact the study director and explain that perhaps your plot does not have as much of the crop left as you had hoped, and ask for permission to collect just 1 sample on the decline dates. And the study director could potentially amend the protocol to allow that. Have I answered the question?
Ken, do you have something to say? And I'm sorry, Geoffrey from Uganda wants to say something regarding this question. So go ahead.
Yes. Thank you, thank you, Adriana. Yeah, I just wanted to add to what Ken said, and that is when you do have a decline trial, that the treated plot should be probably made larger than the untreated plot, because you are going to need to collect samples for a longer period of time. Usually there's 5 collection dates. And something I wanted to add too is that sometimes with the decline trial, that you are allowed to harvest the fruit or the vegetable when they're underripe, but as long as they're full size in maturity. So the first couple of collections, the fruit or the vegetable might be slightly underripe commercially, but they're full size, and so that would be okay. And then it would give you it would stretch out the amount of time that you have to collect that sample. So make sure the plot is large enough, and then maybe the first few samples might have to be slightly underripe, but full maturity size.
Thank you, Joe. I think Geoffrey from Uganda wants to say something. Geoffrey, please go ahead.
Yeah, thank you, Adriana, for giving me the opportunity to comment on this. I think in our experience with the decline study, what Joe has mentioned is really a typical situation you experience at the initial stage of the sampling, that you realize some of the fruits are not fully ripe, and you really need to take that into consideration. And then the decline study, where you're sampling, say, at 5 different intervals, you run into a situation where towards the last stage of the study, you are basically, struggling with collecting the samples. So I think with the guidance given in terms of the actual plot size, my concern was, when you're in the middle of a study, the amendment to the protocol, I think that's why you might not foresee it coming. So I think Ken is right. At that point in time, really, we need to get in touch with the study director so that he can really help with the amendment of the protocol immediately. Because these are things you really don't foresee in actual study when it is to come. And then the other thing maybe I also wanted Ken to comment about is at times when you're running a study which is involving an insecticide, you have a situation where a fungal infection is there, and then you need to manage the crop. So these are things which, again, really you need a bit of care in really dealing with the quality of the crop when it comes to sampling. So those are the things really, from a more practical point of view, you tend to experience when you're carrying out a study. Thank you.
Ken, do you want to say something?
Yes, just briefly. First, to build on what has been said about the decline, the foundation is working with many different crops, and some of them are very new to us who are writing protocols. And if you receive a draft protocol for a decline trial that has a much wider range of sample collection dates, then you know the crop is really available during the field. If it has a very narrow harvest period and the decline trial is spread out over 3 weeks, that is something you should contact the study director about and discuss when the crop is typically harvested. And Joe is correct that you may harvest immature crops if the size is mature, but the study director can choose to make a more narrow period for the decline trial, because there is really no specified collection dates for a decline. That is entirely up to the judgment of the study director, and that can usually be adjusted to match the harvest period for the crop. As far as the question of problems occurring in the field, fungal diseases, or it could be birds attacking the crop and impacting the health of the crop and how much is left. That is the sort of thing that it happens in agricultural trials. It cannot be completely prevented. And if that is happening in a trial you are involved with, then you need to let the study director know what is happening, what measures you would like to take to try and reduce the damage. Sometimes with birds, the study director can permit netting to be placed on top of the certain crops to protect them from birds. With the fungal diseases, if you have a registered fungicide, you may use that to protect your crop, as long as it is not as long as it is not a fungicide that is too similar to what is being tested. And the same goes for the use of insecticides to manage insects that are moving into your plots. But just stay in touch with the study director and with your local coordinator, if one has been named in the protocol, to keep them up to date on the health of the crop in your plots.
Muchas gracias, Ken.
Thank you, Ken. We are really impressed. We reached the 4 hours of the event and we still have 370 people connected. We would love to have more time to talk about all these questions in more detail, but we must finish for the time being. Nonetheless, we revise these questions in the chat afterwards and to respond these through email. If we have enough time tomorrow and there's a time for questions, then maybe we can face some of the questions tomorrow, maybe even opening the microphone so you are not left with the question. We will give you all our contact information, and this foundation is at your disposal. You can write to us and clear any doubts you might have. Eric? Thank you, Veronica. Great idea for the management of the questions that have not been answered. We still have 1 minute here, so please don't keep the questions to yourselves. Write them down in chat, and with certainty, the people from the foundation will be able to manage these questions accordingly. And if anyone still has not written down their name, institution, and country, please do so. We thank you all very much for doing that. It has been a very punctual event with respect to time, and I would like to thank all the different panelists for this. This is a quite long event to do this virtually, 4 hours, but really it has captured the attention of all of us because we have maintained the number of attendance during the whole event. Tomorrow we'll be talking about the different principles, and we'll have some presentations by officials of some countries and consultants of the foundation. And we'll talk about the trial elements, operating procedures, implementation of the study, data notebook, and reports, and storage and conservation of files. Therefore, I am sure that you will all do your utmost to continue with us for this workshop. We remind you that the workshop starts early for these Latin American countries. We'll have some reference times: 8 a.m. New York time, 6 a.m. Costa Rica time, and for some Central American countries, 7 a.m. for the countries in the Andean region, and those participating from Africa. Then give you as a reference, the beginning time would be Kenya time. It's 3 o'clock, 3 p.m. Kenya time. For me, it has been a pleasure to be able to share with you this event. We expect to see you all tomorrow, and I hope you all have a great day. Thank you very much.